Dr. Olak? Dr. Olak? I think there is some network issue with him. Yes, Dr. Olak, we can start. There seems to be some network issue. Dr. Olak? There is some network issue and we are trying to resolve that with the Dr. Olak. So in the meantime, if the participants, if they have anything to interact with Dr. Malik or with me or among themselves, we can do that. So you're most welcome for that interaction and we would love to do that. Yes, Dr. Olak, it is your screen is visible, but your sound, we are not audible, we are not able to hear you. We are not able to hear you. In the audio part, what you have selected for the microphone, I think there is same as system. Just this lower one. No, select a microphone. You have selected the same as system. Just below that. Instead of same as system, select the other one. Let's see if it works.
Hello.
Yes, yes, it is. It is audible. Yes. Join with computer audio. So now I request Dr. Yogita Bansal to continue with her talk. Over to Dr. Yogita Bansal.
So good morning all. Am I audible?
Yes.
Am I audible to all?
Yes, ma'am, you are audible.
Dr. Gulshan?
Apologize to the participants. Sorry, it was very unfortunate.
Yes, you are audible. Please continue.
So very good morning to all participants. Thank you, Dr. Gulshan, for such a nice introduction. So I just came to know that there was some problem with the speaker. So I'm just going to start with today's session. Today is the 5th day of your GIAN course, which is coordinated by Dr. Malik and Dr. Gulshan. I think you all must be very well aware of various types of pollutants and their impact on the environment till date. So the aspect or the area which I am going to discuss today in my presentation is about drug synthesis, how the drugs synthesis of the drug acts as a potential source of the environmental contamination. So now, just hold on. So a drug or a medicine is any chemical substance that causes a change in an organism's physiology or psychology when consumed. Medicines can help to control the things like high blood pressure or high cholesterol. Usually medicines are very commonly taken in our homes to control various kind of physiological diseases. These drugs don't cure the underlying problem, but don't only cure the underlying problems, but they can also prevent some of the body damaging effects. As the world's population continues to expand, the pharmaceutical products, generally drugs and the chemical-based care products have continued to become more prevalent, and their significance in the modern world or modern life cannot be underestimated. So the discovery or the drug development is generally via various steps, that is discovery and development, preclinical research, clinical development, FDA review, and post-marketing monitoring. These are the various stages which the drug has to pass before coming into the market. The major steps is in these drug discovery program is the discovery and development of the drug, which involves the conversion of, which involves the various aspects of science, that is chemistry, biology, and medicine. The chemistry mainly involves the synthesis of the compounds, synthesis of the compound library as it is represented in the picture, that various compound library is synthesized, and through in vitro high throughput screening, the lead is identified, which can further be modified for the better drug. Okay? So synthesis is the heart of the drug discovery and development, and the industry is increasingly demanding the higher quality of clinical candidates and has increasingly demanding the higher quality of clinical candidates and has sought to exploit less druggable biological targets. So these small molecules, they are densely functionalized and have high degree of synthetic complexity is required for the synthesis of these drug molecules. This compound synthesis is considered as the rate limiting step in many drug discovery efforts, because as I told, that synthetic complexity, high densely functionalized small molecules are required. Reactions commonly used in the early stage of development are those that with conditions favorable towards reagents and products having these functional groups. So this is the, I'm showing that how the drug synthesis from the bench or the lab scale is then moved on to, initially moved on to the pilot scale and then to the commercial scale. This is the visual representation of the various stages. So in the pharmaceutical industry, when this drug synthesis moves on to the commercial scale, the pharmaceutical process is involved, then the various pollutants which are generated from this pharmaceutical, there are various pollutants which are generated from this pharmaceutical process. During the last 3 decades, chemical pollution due to synthesis of the drug by conventional ways is exclusively increasing at an alarming rate. Plants synthesizing the drugs and their products emit many volatile organic compounds and other pollutants in the environment via air emissions and wastewater and solid waste. The wastewater, now due to the purity concerns, most of the solvents used in the pharmaceutical manufacturing activities are not recycled for reuse. As a result, the pharmaceutical wastewater contains many hazardous chemicals, including varying concentrations of organic solvents. Some priority pollutants in the pharmaceutical waste include isopropanol, acetic acid, ethyl acetate, ethanol, N-heptane, THF, that is tetrahydrofuran, methanol, and toluene. Now, various air emissions, predominant volatile organic compounds are the solvents because they are mainly present in the predominant volatile organic compounds like acid gases, halogen acid, sulfur oxides, and nitrous oxides, which are emitted from the production plants. They cause pollutions. So depending upon the type and the quantity of the reactants and the solvents, some plants may be negligible sources of volatile organic components, while the others are responsible for high source of pollution due to these components. In addition, all types of equipments which are used in these pharmaceutical processes, they have potential to emit air pollution. Reactors and distillation operations are major source of pollution due to air emission. Now coming to reactors, the emission of air containing these volatile components during the reactor charging, hazardous solvent, evaporation, during the reaction as a byproduct gases, overhead condenser venting, uncondensed volatile organic components during refluxing, purging, vaporized volatile organic components remaining from the solvent wash. Now coming to the distillation operations, which are again used to distill off for the purification of the organic compounds, mainly distillations are used. So the magnitude of the polluted volatile organic components depends upon the operating parameters of the condenser, type and the quantity of the organics being condensed, and the quantity of the inerts involved in the reaction. Now solid wastes, raw materials or the products, used solvents, reactants, reaction residues, filtered media, filter cakes, used chemical reagents, dust particles from filtration or air pollution control equipment. Raw materials from the packaging wastes, laboratory waste, spills, and waste material generated during the packaging of the formulated product also act as the source of the solid waste. Now, drug synthesis, as it is the drug synthesis, what is drug synthesis? It is the conversion of the existing molecules into the other useful molecules. So that the useful molecules means the drug must be able to be absorbed into the body. It should have good absorption. It should be very well distributed to reach its site of action, right target. And we must know the metabolism of the drug, that how the drug gets metabolized, what are the metabolites of the drug, as metabolites of the drug are also a very potential source of the pollution. In the pharma drug pollution, mainly sometimes the drug metabolites are more pollution causing than the individual drug itself, than the parent drug. The metabolites of the drug, how it will be metabolized is very important to be understood before the discovery or before the identification of the drug. So we must also know that how drug gets excreted out from the body, and the human excreta is again a very potential source of pollution, mainly water pollution, soil pollution. We must understand the toxicities of the drug also before synthesizing the newer molecules. Now, this drug chemistry, what are the various, what steps are involved, how we are converting a drug into a useful molecule, how we are converting an existing molecule into a useful molecule is by either converting a little molecules or the simple molecules into the bigger one. As previously told, that drug is a complex, having complex functional groups. So little simple molecules can be converted into bigger molecules by a synthesis. Then synthesis of the unnatural products from the natural products. Sometimes we isolate the phytoconstituents from the plant products, which are individually, they are not having that potency which is required. And we can modify via various chemical methods. Different introduction of the groups can be done on those natural products. And then the synthesis of the drug is carried out by modifying the natural products. And modification of the existing structure. The example, for example, morphine is one of the natural products which is isolated from poppy. And morphine, because it, although it is having a pharmacological use, but it is also having a drug abuse. So those various structural modifications have been carried out in the morphine, or the structure is broken and structural modifications are carried out for the development of newer molecules or newer synthetic molecules, which are more capable of having the analgesic effects and they are of less abuse. This is one example which I am giving where the synthesis of unnatural products from the natural products can be carried out. So now the various steps which are involved, how this chemical synthesis, what are the methods and strategies which we follow for the drug synthesis? The very first is the formation of CC bond, carbon-carbon bond, because the drugs are the chemical compounds. So a carbon-carbon bond, various reactions can be involved in the formation of the carbon-carbon bond. I'm showing you very few examples. There are numerous number of reactions which are involved in the chemical synthesis, in the drug synthesis. Very commonly used, some of the reactions are aldol reaction. Aldol reaction is a reaction in which beta hydroxy unsaturated carbonyl compounds, functional moieties are generated by utilizing this aldol reaction. Example I am giving is the synthesis of Taxol. Taxol is an anti-cancer drug which is synthesized utilizing this aldol reaction. Next reaction is Diels-Alder reaction. Diels-Alder reaction is again a cyclization reaction in which a cyclic cyclohexene can be obtained, cyclic structures can be obtained, and because this steroidal drugs and cortisone, they are the cyclic moieties. Steroidal drugs are having tetracyclic rings or pentacyclic rings. It is a cyclopentanoper hydrophenanthrene. This is the chemical name of a steroidal nucleus. So because it is having the cyclic rings, these rings can be formed or synthesized via utilizing the Diels-Alder reaction. Very commonly used steroidal drugs are progesterone, estradiol, diethyl estradiol. Diethyl estradiol is very commonly used and is a very potential source of the aquatic pollution, water pollution. You must have till now heard from any speaker about the pollution caused due to this diethyl estradiol. Next kind of the reaction is addition of Grignard's reagent to a carbonyl group. I will give you an example of addition of a Grignard reagent to a carbonyl. How this Grignard reagent is a magnesium containing reagent. So this is a metal containing reagent which can again act as a potential source for the environmental pollution. Example next is the Heck reaction which is involved in the synthesis of a naproxen, which is a very commonly used anti-inflammatory drug. So again, the various steps involved in the synthesis of naproxen utilizing this Heck reaction may act as, at the different levels, may act as the source of the environmental pollution. Other very commonly used reactions for the formation of CC bond are Michael reaction and Wittig reaction. These are commonly used in the production of drugs because they are mainly involved in adding the double bond near to an aldehyde or a ketonic group, which is a very commonly used functional moiety in the various drugs. The next kind of method or the strategies involved in the drug synthesis is functional group interconversion. One of the functional group is converted into the other functional group. This can be done by utilizing various nucleophiles or electrophiles. These are the reagent, nucleophilic reagents or electrophilic reagents. And the various interconversions involve oxidation, reduction, substitution, elimination, addition, and these are various kinds of reactions which are involved in the functional group interconversions. The oxidation reduction reactions mainly involve the usage of metallic compounds and which can again act as the potential source of environmental contaminant, which can act as the potential environmental contaminants. So this is the, simply I am showing you various examples of substitution, addition, and elimination. These are some of the interconversions which I have shown here by utilizing either the nucleophile or the electrophile. So these reactions are very, very commonly used in the drug synthesis. Substitution, addition, very common reactions used in the drug synthesis. I'm giving you an example of a drug, ibuprofen, which is an anti-inflammatory, non-steroidal anti-inflammatory drug, which is used for its analgesic property. And ibuprofen, you must all be knowing that it is very commonly used anti-inflammatory and analgesic drug. This synthesis of ibuprofen involves mainly these steps. These are the 5 steps which are involved in the synthesis of the ibuprofen. These are the reactions I had highlighted that the very first step is involving the synthesis, involving Friedel-Crafts acylation, and in the later stages we are involving Grignard's reagent reaction. Now this Friedel-Crafts acylation is involving isobutyl benzene, and acetic anhydride is used to create the para-isobutyl acetophenone in the presence of hydrofluoric acid, which is the acid which is used as a catalyst in this particular reaction. Then this is reduced with the presence of sodium borohydride. So borons are involved, boron hydrides are involved in this reaction. Next, in the presence of Grignard's, utilizing the Grignard's reaction in the presence of magnesium and carbon dioxide, this ibuprofen, skeletal of the ibuprofen is synthesized. Now, these are the reactions which I had discussed, that which are utilized in the synthesis of ibuprofen, Friedel-Crafts acylation reaction, and this Grignard's reagent, which is an organometallic reagent using magnesium halides. And just hold on, I am trying to rectify it. So this we were discussing about the reactions. So now, as we had seen that the various things which were involved in this synthesis of ibuprofen were acetic acid, which facilitates, now this toxic effects of acetic acid are that it facilitates the formation of ketone bodies, causes hypotension and arrhythmia. This causes extreme eye and nasal irritation, conjunctivitis, permanent corneal opacification, and excess of prostate cancer is observed among the chemical plant workers, which are exposed to both this acetic acid as well as acetic anhydride. Acetic acid is mainly formed by the hydrolysis of acetic anhydride, which we had used in the reaction. Now, the Grignard's reactions which were used in the synthesis of ibuprofen, this also present the challenges, particularly on the scaling up. From the safety standpoint, the initiation step is often hard to control and is strongly exothermic. In some instances, research scale chemistry has been recently carried out using continuous flow methods to alleviate the exothermic hazards. This has been now observed in the pharmaceutical industries that to control the pollution, these are some of the methods which are used, that rather than batch scale production, the continuous flow methods are mainly used to alleviate the exothermic hazards or to alleviate the other pollution effects of the particular processes. Furthermore, as the solvent involved in these reactions are mainly diethyl ether and tetrahydrofuran, which have low boiling point, and they are capable of forming peroxides. Peroxides are again very hazardous as well as detrimental to the human health. The ecotoxicity studies of acetic acid shows that it is harmful to the aquatic life, and high concentration produces pH levels that are toxic to oxidizing bacteria and inhibit the oxygen demand. I think I'm facing some problem in shifting the slide. So effect of boron on the organisms in the environment is bacteria are much less sensitive to boron, as I have shown previously that boron hydrides were used in the synthesis of ibuprofen. As bacteria is much less sensitive to boron compared to other chemicals, protozoa are somewhat more sensitive. Algae, for which boron is essential nutrient, and invertebrates have low boron sensitivity. Fish are the most sensitive species to boron. When animal absorbs large amount of boron over a relatively long period of time to food or drinking water, the reproductive organs are affected. When the animals are exposed to boron during pregnancy, their offsprings may suffer from birth defects or delayed development. The other effects of boron on plants is also seen, that it is mainly affecting the plants because there is a narrow margin between a boron deficiency and excess boron uptake, leading to the toxicity. Irrigation water, which is containing boron, suppose when a drug is synthesized and the boron enters into the aquatic environment, and that water is then utilized for irrigating any farms, then that irrigating water, which is containing boron, is act as the main source of causing boron toxicity on the agricultural land. This is the another example which I am showing for the synthesis of ivermectin. Ivermectin, you might have studied that it is a broad spectrum anti-parasitic agent, which was very commonly used in the previous day of COVID. The ivermectin was very commonly administered to the COVID patients, and it increases the acute immunity. They have been used in the agriculture and horticulture for the protection of fruits also, cotton, vegetables, and ornamentals. Now, the synthetic reaction of ivermectin involved the usage of phosphorus, phosphine, rhodium chloride. Okay? Again, the utilization of metallic catalysts, and these are extremely toxic to aquatic crustaceans and highly toxic to the free living nematodes, particularly cladosarians, and even at the concentration of 0.001 to 25 nanograms per liter. Next example, which I am going to take, is of the diclofenac, which is a potent anti-inflammatory drug. And thousands upon thousands of the vultures had died in the last 2 decades due to the diclofenac toxicity. This is the synthetic scheme which is utilized for the synthesis of diclofenac. Again, I am showing that usage of copper, usage of lithium aluminum hydride, usage of cyanides in the synthesis of diclofenac. So all these copper, lithium, these are the various toxic effects of lithium and copper, which I have shown in this slide. Copper can travel to the great distances in surface water and can get either suspended on the sludge particles or as the free ions. Copper does not break into the environment and therefore can accumulate in the plants and the animals. And we can see that because only the limited number of plants have a chance to survive in the copper-rich soil. Therefore, there is very less plant diversity near the copper disposing factories. Copper can also interrupt the activity in the soil, and as it negatively influences the activity of microorganisms and earthworms. Consequently, the decomposition of the organic matter may seriously slow down. Animals absorb copper from farmland polluted with it, which is damaging to their health. Mainly sheep suffer from a great deal of copper poisoning because the effects of copper are manifesting at fairly low concentrations. Now, lithium reacts with nitrogen, oxygen, water vapor in air, and consequently, the lithium surface becomes coated with mixture of lithium hydroxide, lithium carbonate, and lithium nitride. Lithium hydroxide represents a potentially significant hazard because it is extremely corrosive. It affects the growth of water organisms also. Now, the aluminium also causes the cause of toxicity to the freshwater biota. It affects vascular plants, and freshwater invertebrates disappear in the acidic waters as a response to low pH and aluminium. So these were some of the effects of few drug synthesis. I had discussed the ibuprofen, ivermectin, and diclofenac. Now, this drug synthesis, as discussed, is involved in the synthesis of. It is involved in the drug synthesis is involved as a potential source of pollution. Now, another methods which are also used at the industry level for the in the pharmaceuticals, for the development of pharmaceuticals, are fermentation and extraction. What is fermentation? Fermentation is mainly industrial microbiologists use this fermentation method for the production of antibiotics. Now, in this fermentation, a source of microorganism is grown in large containers containing a liquid growth medium. A particular oxygen concentration, temperature, pH, and nutrients are closely controlled in this method, in this fermentation. Now, this I am just showing you this, that how a bacterial culture is initially started in a small test tube filled with the growth medium, and then it is expanded in a nutrient-rich medium and continuously shaken. And then how this can be utilized for the commercial scale production via mechanical mixing and for the commercial scale production of various antibiotics. Next method which is used in the drug development is extraction. The pharmaceutical processes involve extraction of the organic compounds for their use as medicines from plant materials or animal tissues. Extraction is used to separate the liquid mixtures by taking advantage of differences in the solubility of the mixture components. A solvent that preferentially combines with only one of the component is added to the mixture. The resulting mixture consists of an extract and a raffinate, that is containing a residual phase. The various solvents are used in this extraction process like benzene, toluene, chlorinated and polychlorinated solvents, and other organic solvents like DMSO, DMF, petroleum ether, diethyl ether, acetone. They are used in great quantities in these extraction methods, and they can cause environmental problems. Long-term exposure to solvents and air pollutants can lead to deleterious effects on respiratory, hematology, and thyroid functioning. So these are some of the effects of organic solvents, which I have discussed. Many occupational diseases caused by the chemicals results from breathing air that contains harmful substances. Now, exposure to these hazardous materials can be acute or chronic. Acute exposures generally refers to single dose. High concentration exposures over a short period, while the chronic exposures involve repeated or continuous exposures over long periods. These exposures may have acute, immediate effects, or chronic, or long-term effects. These are the various effects which these organic solvents can have on the respiratory tract, on skin, like asthma-like disorders can be there. Skin, they can cause drying, cracking, reddening. In the eye also, they cause reversible or the permanent eye damage. Is very rare, although it will cause the eye irritation. Liver damage when exposed to carbon tetrachloride or other chlorinated hydrocarbons or ethanol. The liver damage can occur. Carbon tetrachloride, trichloroethane, petroleum distillates like jet fuel, turpentine, gasoline. These are again toxic to kidney. And these are some of the other effects which these solvents may cause on the different parts of our body, that is cardiovascular system, nervous system. And hematological effects can, for example, exposure to the solvents like benzene, they decreases the RBC count and hemoglobin level, causing anemia. So this all I had explained on the small scale, that the examples also which I had taken was ibuprofen. I had given you the various steps which are involved in the synthesis. Now, pollution from the production of these pharmaceuticals has not been previously considered to be significantly contribute to the release of the drugs. Initially, it was thought that drug pollution is mainly due to the consumption of the drug, that is mainly from the excreta or from the domestic waste. Mainly, initially, it was considered that main source of drug pollution is mainly from the excreta, but or due to the improper disposal of the chemical waste. But pollution from the production of pharmaceuticals is now becoming very significant, and recent investigations have shown that Indian and Chinese production units have shown that certain production sites are contributing. I will discuss the case study of Hyderabad, as it is the hub of drug production. We will discuss it later. Here I am just showing you a few examples of the drugs which are when move onto the soil or water, how they are having the ill effect on the environment. Initially, I am discussing the discovery of estrogens that in the sewage effluents, they cause feminization of fish in late 1990s, and it has exponentially increased with the time. Sewage effluents and receiving rivers are thereafter the prime focus in the pharmaceuticals. So the proper treatment plants should be there for controlling the sewage effluents. The discovery of diclofenac residues in the cattle carcasses as a cause for the vulture population collapse in India and Pakistan. So diclofenac was the major cause of cattle carcass. In 2007, the first in the series of papers was Hyderabad. This is the case of Hyderabad again, Patancheru, which I will discuss in detail. The alarm from an environmental perspective first became apparent in 2004, where the incidence of 14 gene resistant to first line anti-bed bug infestation pyrethyroid agents were identified in 21 distinct bed bug species from different sites across United States of America, especially in the Midwestern region. The RNA interference was found to hinder the gene expression in these bed bugs, and these genes were blocked. The bugs were susceptible to pyrethyroid again. Minimata disease was observed in human consuming fish and shellfish contaminated by wastewater discharge from the chemical plant containing methyl mercury. So this is the pictorial representation of how these drugs or the effluents are moving onto water or landfills. And this is when the drug reaches to the aquatic system, either from the pharmaceutical production or from the livestock and the human excretion, it gets transformed in the aquatic environment via various processes of photodegradation, bioaccumulation, biodegradation. The most important are the factors which involve the transformation of these APIs, that is active pharmaceutical ingredients, are inherent physicochemical properties of pharmaceuticals, light availability, temperature, pH, oxygen availability, and microbial communities which are present in the aquatic environment. These are some of the documenting studies on the treated industrial effluents, waterways, river sediments. These are showing in India specifically. I have highlighted that the presence of the salicylic acid, which is an anti-inflammatory, presence of many fluoroquinolone antibiotics in the groundwater or in the aquatic environment. Now, this is the case study which I want to discuss here about Hyderabad. The environmental presence of APIs and their association with the drug resistance mainly with gram-negative bacterium in Hyderabad. This is a major production area of global bulk drug market. Water samples from different areas around Hyderabad were collected, and from the vicinity of what were collected from the direct environment of bulk drug and manufacturing facilities, vicinity of 2 sewage treatment plants, Musi River, which is flowing near Hyderabad, and habitats in the Hyderabad and nearby villages. Samples were analyzed for 25 anti-infective pharmaceuticals with liquid chromatography and mass spectrometry for the gram-negative bacteria using chromogenic culture. You might have studied about this liquid chromatography and mass spectrometry. These are the techniques which are used to check the levels of pollutants in the environment in the various samples. Insufficient wastewater management by bulk drug manufacturing facilities lead to unprecedented contamination of water sources with antimicrobial pharmaceuticals, which seems to be associated with the selection and dissemination of carbapenemase producing pathogens. The development and global spread of antimicrobial resistance present a major challenge in the pharmaceutical. This is the image of the area in Hyderabad, which is a polluted sewer because of the disposal of the industrial waste. Just hold on. Has stopped. So sorry for the inconvenience. Actually, there was some interruption. Screen sharing was stopped. So I was discussing about the different samples were collected from different sampling sites around Hyderabad. And this is the data of the presence of different kinds of antimicrobial agents which are present in different samples around Hyderabad: fluconazole, voriconazole, moxifloxacin. These are present in very high concentrations, up to 694, 2500 micrograms per liter. So this increased concentration of antibiotics in these samples may cause the drug resistance. Very major problem which is coming is that of drug resistance because of this. So currently Hyderabad accounts for approximately 40% of the total Indian bulk drug production and 50% of the drug exports. The pharmaceutical industries and their exports are expected to grow 20% annually. Despite decades of campaignings of local NGOs and legal action taken to the highest Indian courts, the pollution of the surroundings of manufacturing plant has not been reduced. Even the Supreme Court has issued that proper disposal of the wastes, treatment plants should be there before disposing of the waste, but still they are not following because of corruption. So now I am discussing the effects of the various anti-neoplastic drugs when present in the aquatic system. The mixture of anti-neoplastic drugs cause more potent DNA damage to the non-target cells, and even at low concentrations when compared with the parent drug itself. EC50 value of bleomycin and vincristine was found to be in the range of less than 10 milligrams per liter. Platinum-based drugs such as cisblastin or carboplatin are commonly present in the hospital effluents, and they are considered to be highly toxic to the aquatic organisms. Cyclophosphamide and fluorouracil have been reported to cause the mutagenic effects in tadpoles. Fluorouracil, imitinib, and cisblastin are the most potent drugs to cause transgenerational effects on the certain aquatic species, and many other the native or the parent compounds as well as the byproduct form of these drugs can induce the adverse effects on both aquatic species and human life. Examples: direct physiological effects, genetic material damage, and immune system damage. This is the that bioaccumulation and biomagnification, which I have previously told that may lead to the high levels of anti-neoplastic drugs in the aquatic environment. Several studies have reported the toxicity of various anti-neoplastic drugs on different aquatic organisms. Now, this is a very important case study which I will like to discuss in this presentation. It is a case study of an active pharmaceutical ingredient which is produced or whose production is carried out in Basel in Switzerland. These are the very good methods, studies which are carried out nowadays to prevent the environmental pollution from these pharmaceutical industries. That is, the life cycle inventories of various products are being studied. There are many reports publishing the life cycle inventories of various drugs. There were recently a report which was publishing the life cycle inventory assessment of 20 analgesics, and one more study was there which I also wanted to discuss, but because this shortage of time or prepone of the seminar, I'm not able to now discuss the case study of one more drug. But I will discuss the case study of this. This is the substance A because of the name of this drug is not revealed here, but the various steps which are involved in the synthesis of this substance A are: this is the multi-step reaction which is utilized for the synthesis of this substance, and some of these are shown here. Now, the various parameters which were studied for this, the assessment was the cumulative energy demand. This cumulative energy demand is a method which assesses the primary energy demand of the production. This is a good indicator of chemical production, in particular, as oil and gas are both the main energy source and main material feedstock for chemical production. Now, global warming potential, this is the another parameter which is evaluated, which addresses what may be the most discussed environmental issue of our time, and many studies require global warming potential. The other indicators which are used to assess various environmental impacts are Eco Indicator 99, Ecological Scarcity 2006, and it is TRACI, TRACI is tool for the reduction and assessment of chemical and other environmental impacts. Now, these are the various, these are the results of the life cycle impact assessment of the pharmaceutical A. So this is the various values which were observed when the life cycle was assessed for this substance A, that these GWP, CED, these are the values, and these eco indicators which were observed to be significantly high in the production of this substance. So this is the graphical representation, which is showing the relative impacts of energy production and use over the production of A for CED, GWP, EI99 and ES 2006. And this is shown that it is increasing as in the process steps starting from cyanoacid acid was one of the reactant which was used, initial reactant which was used in the synthesis of substance A, and it is observed that all the parameters are significantly increasing during the process. When the production of this active pharmaceutical ingredients was compared with oil refining from the bulk chemicals, fine chemicals, this is the estimated total waste in the tons per annum, which was calculated for these different kinds of the pharmaceuticals. It was observed that because the bulk chemicals, they are mainly requiring the 1 or 2 chemical transformations in comparison to the pharmaceuticals, which are requiring more than 6 transformations. So they can act as a more potential source of the environmental pollution. The analysis of the requirements and impacts of A's production were compared with the analysis of 78 basic and mass-produced organic chemicals. Basic organic chemicals means very simple chemicals which are required in the laboratories, which are required in the various processes. When the production of a pharmaceutical ingredient was compared with the production of these basic organic chemicals, it was observed that average CET, cumulative energy demand, of basic chemical production is only 5% of the cumulative energy demand of A synthesis. Matlab, the synthesis of pharmaceutical ingredient A is requiring very high energy demand in comparison to the basic chemical production when it was analyzed on the per kilogram of the product. Moreover, the average basic chemical production impacts are only 4% for the GWP, 6% for the ecological indicator, that is E199, and 3% of ES 2006 when compared to the production of A. And moreover, the TRACI percentage also dropped to less than 1%. That is, the simple organic chemicals, they are having very less impact. The production of simple organic chemicals is having very less impact in comparison to the production of the pharmaceuticals. Now, what is the reason of this? Why pharmaceuticals are having more impact on the environment? Now, the pharmaceuticals are more resource intensive productions. So rather than the simple chemicals, they are resource intensive productions rather than the simple chemicals. Now, how they are resource intensive? The production costs are generally outweighed by the cost of R&D or the marketing. The economic incentive to optimize the production is lower than in the production of the other chemicals. There is very less time to increase the efficiency of the processes, as the time to market is crucial for the pharmaceuticals. So in the case of pharmaceuticals, because already drug discovery and development, the stages which I discussed initially, that drug discovery and development, then preclinical evaluation, then clinical evaluation, and then FDA approval, all these processes is a very long process. Any drug to come into a market takes a very long time, around 12 to 15 years. It is a process of around 12 to 15 years. So because there is the industries or the researchers, very less time is taken to increase the efficiency of the process, because they want, the industries want the drug to come to the market as early as possible. So the pharmaceuticals often undergo, moreover, the pharmaceuticals often undergo formulation and purification processes after production to ensure the product purity and the pharmacological function is maximized. Product purity is very crucial in these cases because it is of human use and the pharmacological function is to be maximized. So these steps are also very energy and resource intensive. So they are further requiring energy demand, and resource demand is very high in the case of pharmaceuticals. So they are very potential source for the drug development. So in the last, I will discuss the how to manage the, just wait, again stopped screen sharing. I don't know what problem is coming, just wait. So in the last, we will discuss, as till now, we had discussed that how the pharmaceutical disposal or the manufacturing of the pharmaceutics is causing a problem in the environmental pollution. So how to manage this environmental pollution by this, from this pharmaceutical disposal, and what are the environmental standards which are to be maintained? The various practices, good manufacturing practices are to be followed by these pharmaceutical industries. These are the principles and procedures which are required to maintain the consistency and quality standards of the therapeutics, covering the manufacturing and testing of these APIs, diagnostics, and food world, primarily in the developing world. Proper disposal of the unused medicines, well-equipped sewage treatment plants, education for the rational use of drugs, and reinforcements of the biodegradable products should be carried out. The principles of green chemistry could be applied to the environmental pharmacology. This green chemistry we will discuss again in one more lecture. Now, what is this green chemistry? The various principles are prevention of the waste products, maintaining the economy of the atoms, non-hazardous synthesis, synthetic procedures which are to be followed should be non-hazardous, metamorphosis of intermediately used chemicals, reusable feedstocks, reducing the number of toxic metabolites, using recyclable catalysts, and real-time analysis for the pollution prevention. This is a very important step which is to be followed in the manufacturing processes, that is real-time analysis. At every step, real-time analysis is that the reactions or the processes should be monitored while they are carrying out at every step. So that what any gas leakage is occurring or not occurring, well, the real-time analysis will prevent the environmental pollution. So eco-pharmacovigilance is the another area which is very commonly used nowadays. It is the defined as the principle and the practices assuring the safe disposal of byproducts and waste materials from the manufacturing plants, as well as the proper handling of the drugs and syringes in the healthcare premises and sewage plants. Various practices ensuring the risk assessment and eco-friendliness of the drug can be adopted throughout the life cycle of the drug, especially after the launch of the new product. These includes ensuring proper disposal of the effluents from the manufacturing and environmental wastewater management, monitoring of the environmental risks after the launch of the product, assessing and managing the risk factors across the life cycle of the drug. And this can be carried out by assessing the life cycle of the particular drug using environmental risk management plans and resources. Discerning and monitoring after the risk is identified, keeping a universal EPV perspective, and increasing the transparency and availability of the environmental data for the medicinal products. So thank you. Any questions? This presentation actually, because it was pre-planned, I was not able to, I was into the process of preparation. So any questions?
Thank you, Dr. Yogita, for your nice talk. If any participant has any query or any feedback or any suggestion, kindly feel free to ask. Yes, any suggestion, please? Any query? Am I audible?
You are audible.
Thank you, Dr. Jitendra. So if there is no query from any participant, so we can move on to the next session because we are running short of time. So because of this technical snag, we had to shift the sessions, and because we started around 40 minutes late, so we have to make up this time. And now the next session will be by Dr. Jitendra Singh Aulak on the theoretical concepts of solid phase microextraction for sample enrichment, and which is an excellent pre-concentration technique. Uh, so after his this talk, he will take us to his laboratory where he will demonstrate how solid phase micro extraction works, and how the sample enrichment is done, and its practical aspects he will share with us through demonstrations, which will be very beneficial. Otherwise, we had planned it in an offline mode for the last 2 days, but as per the instructions from the funding agency, we had to shift to a totally online mode. So, and because of this, we have to also this day shift our lunch hours, because the lecture will be of around 1 hour duration, and 2 hours will be for the demonstration. So we will be shifting the lunch hours from 1 to 2 to 2 to 3. So kindly bear with us. And now I request Jitendra to continue with his presentation. So if there is any query or any issue to any participant regarding this shift, he or she can share his or her concern. We have to shift it because that demonstration session is for 2 hours and we cannot split it into 1+1. So that's why that is a compulsion. So thank you so much for your consent. Okay, so Dr. Jitendra, please go on.
Thank you, Dr. Gulshan, for the nice introduction that you gave in the morning. And I apologize for the inconvenience that is caused to all the participants. And this was all because of some internet issues, which was beyond my control. But hopefully now everything will go right. As an sample preparation and sample purification technique, is it visible, sir, now?
Yes, it is visible.
Okay.
There is just one issue, I think, which we may face if we shift it to the full screen mode. Then whether the screen will shift or not, that we have to see. Just shuffle your slide, please.
Okay.
So just first make it into the full screen mode, and then share the full screen share, window.
So I will again share. So screen. So now it is okay, sir?
We are seeing your desktop. Okay, just start from here. If we will face any problem, then we will see. Yes, it is visible. Your slides are visible.
presentation today will be the solid phase extraction for sample enrichment. Solid phase extraction, for detailed studies, if you want, we have the two books which are very commonly important that we should understand the need instruments.
There is again some audio problem, Dr. Chitender. Your audio is, you see, breaking in between. Slide transition is perfect.
Slide okay, sir?
Slide
The audio will be okay, sir.
audio
I think
is in between somewhere. It it breaks. I don't know. Anyway, we can't do anything.
Okay, I will try it best.
Okay.
My best, sir.
Sure, sure.
So sample preparation is an important step focus is needed on sample preparation is because the time that is spent on the sample preparation is the maximum time, and if we are able complete analytical procedure. So the sample preparation is very important. The main objectives of sample preparations are to remove the potential interference.
Hello. Sir,
To
am I audible?
pre-concentrate the
Hello?
analytes more suitable forms. Yes, sir.
Sir, voice is not coming continuously. There is a lagging in voice. I don't know what's the problem because in between voice is not coming.
Change the room also and everything that is possible here in the department.
Half the statement is listening and after that it is not coming. Others can also tell maybe if there is problem at my end, others can also tell.
Yes, we are facing some intermittent problems with audio. There's actually some network connectivity problem is there. That is the only reason. Let's
I will continue.
Let's continue. Let's see if there is
You can ask me problems, any questions that at the end also.
or you can post your questions in the chat box also.
Yes. Amount, then there is dire need that we should have it in higher concentration so that analysis can be performed. We need if the suitable method is not available for an if we'll see this figure here, the sample pre-concentration sample enrichment means the same amount of analyte which was present in very bulk amount or bulk sample is enriched in a small sample. So how this all procedures are done by your solid phase extraction, we will see one by one. Extraction is a method of sample preparation.
Dr. Jitendra, we are having a continuous problem of this audio. I think without audio, without you see consistent audio, the purpose will not be solved. So how we can fix it? I don't know this one. Is it possible that we can shift the site from university to some extra university site?
It is possible, or I shift my computer to the lab, I think, but there was certain noise there, so I avoided that.
The noise, if the network works there well, then we don't have any issue. Noise we can control at our end, because
Okay, then I will go to the.
we can request the students to keep shut over there.
Done, but what about the lab?
Yeah, lab part that we have to.
I wanted to show the lab.
Is it possible to use the hotspot of some phone? If whosoever is available in your building, if he has having a good phone connection, network connection in his or her phone, then can we use that hotspot? Because we will be having a 2 hours time to fix this issue by that time, because Dr. Hemendra Bharti will be taking a session up to 2 in the afternoon. Maybe we can ask him to curtail it to 1:30. Or alternatively, it's a 11:45. We can have a break now and we can join after, let's say, half an hour. And we can take snacks instead of lunch this time.
This can also be
So I
done.
just want to hear, feedback from the suggestions from the participants, because it's more important to hear from the
Actually-
participants what solution they can have. Because the problem is there, and because without audio there is no, you see, fun in continuing with this thing. So Dr. Hemendra Bharti has joined, and we will be sharing his screen soon. In the meantime, let me introduce Dr. Hemendra Bharti to all of you. Dr. Hemendra Bharti is Associate Professor and currently the Head of Department of Zoology and Environment Science in Punjab University, Patiala. He is a very dynamic and learned academician and very active researcher in the field of ecology, evolutionary biology, and the conservation, environment conservation, and phylogenetics, molecular phylogenetics, systematics, and chemical ecology. So he is a very progressive scientist, and he's recently designated with the duty of being a director to Center for the Conservation of Ecology in Punjab. So he's doing great efforts in conservation of our ecosystem, and he will be talking about the particular effect of pollution on the marine systems, marine organisms. So I welcome Dr. Bharti to share his views and share his talk with the participants. Over to Dr. Bharti.
Yeah, thanks a lot, Professor Gulshan, and thank you for those nice words.
And also, I want to thank you in advance that you have preponed your schedule to save us from this technically unsavory situation. Thank you so much.
Hello.
Yes, Dr. Bharti, you are audible.
And screen is visible.
Yes, it is visible.
So okay. So good noon to everyone, and
Dr. Bharti, we are again having some audio problem. We are again having some audio problem, not the video. Audio problem is there. No, not audible. My God.
hello.
Hello, yes, Dr. Bharti.
Audible now?
Yeah, now it is audible. Somewhere in between the voice breaks. Let's try for 5 to 10 minutes. 5 minutes. If it continue, if the voice continues, then it's okay.
Still, we have glitches and flaws. Hello.
Yes, yes, Bharti. There were some technical glitches, and glitches are always there. So
Yes, the screen is visible now.
yes, yes, it is visible very much.
Okay. So
फिर गया. In audio problem. I'm not hearing you.
oh,
Doctor, sir, he is audible.
He's audible to you? Is there any other participant who is facing the same problem? It is not audible. Dr. Renuka Das said not audible. Then हाँ जी डॉक्टर साहब नहीं बन नहीं रही कर रही है okay chalo let's hope the technical issue is now resolved and it will I hope it will not sustain now, you know, and so we can start the session, right?
Yes, we can, sir. Let me check it out. I will switch off the video. If it is needed, then we can.
हाँ नहीं आप वीडियो भी ऑफ रखो अब ना उसमें फिर वो नेटवर्क का इशू आ सकता है.
So if you don't mind my.
कोई बात नहीं फिर आप
Just
कर लीजिए सेटिंग अपनी कर लीजिए सेटिंग अपनी नो इशू.
Just 2 or 3 minutes में एक बार. Enrichment. Solid phase extraction is basically a sample preparation technique which is far better and far environment friendly as compared to the traditional liquid extractions. Solid phase extraction technique has revolutionized the sample preparation because it can be automated also. If you want to have good details about the solid phase extraction, the good books are available in the market. As the solid phase extraction is a sample preparation technique, so it is very important that we should understand the need or focus on the sample preparation. As we know that for any sample preparation, and if we look into the chances of error, it has been found that the maximum chances of errors are there in the sample preparation process, followed by some other processes that we analyze based on the instrument or integrations, but the maximum time of or maximum amount of error that are present in sample preparation. And solid phase extraction works into this sample preparation because it is possible to have a sample cleanup much quickly here, and also the sample enrichment is done. Also, if you look into the time spent in any analytical process, the maximum time of the total time required for analysis is spent on the sample preparation. So if we are able to automate, or if we are able to do some modifications in sample preparation, then it will be a great help. And SPE is a step towards that. And also, SPE finds very easy to be automated or to be used in line with HPLs. Offline is also there, but inline can also be performed. I will show you how it can be done. The main aim of sample preparation is to remove the potential interference, and with SPE, it will be quite easy. We will see later. Pre-concentration of analyte, this is also the objective of sample preparation. And with SPE, this pre-concentration is achieved very easily. Another approach, another requirement is the modification of the sample analytes for detection and separations. We will see that SP will be a great help for that. And some challenges that we have: use small initial sample sizes, improve selectivity in extraction, facilitate automations, minimize the amount of glassware and organic solvents to be used. So with SPE, the amount of organic solvent that is required, that is also reduced. What solid phase extraction? A basic introduction to that. In solid phase extraction, we have a solid phase which is packed in the cartridges, packed in these barrels, and we are able to extract the analytes which are present in aqueous phase into this solid phase. And it is slightly different from when we say liquid-liquid extraction, where we have two immiscible liquids, and if we want to extract analytes from water or any environmental sample, we have to shake that sample with organic phase, and a lot of organic solvent is needed in that case. Also, there are problems associated with those liquid-liquid extractions. Solid phase extraction is a method of sample preparation that can concentrate and purify analytes from the solution by sorption onto a disposable solid phase cartridge. On a disposable solid phase cartridge, followed by elution. We'll see what are the different steps which are involved in the SP, followed by elution of analytes of interest with solvent appropriate for the instrument analysis. The mechanism of SAP involves reversed phase, normal phase, and ion exchange. All these processes are similar to what we see in HPLC. In HPLC, we have, or in general chromatography, we have reverse phase, normal phase, ion exchange. Here also we have all these retention process, but in case of HPLC, what we want is, we want the analytes to pass through the column bed. But in SPE, we wish our analytes to be retained on the SPE cartridge. SPE procedures are available in various forms. It can be a disc or a cartridge or a syringe barrel. The most commonly used SPE system is the syringe barrel. This syringe barrel consists of a frit at the bottom, frit at the top, and in between
the solid phase or sorbent is packed. These sorbents can be of many types. It can be silica based like C8, C18 and phenyl columns, or it can be polymeric, molecularly imprinted based polymers, or it can be your MOFs, which can be packed here. So there are various possibilities of having sorbent inside the cartridge. This barrel is made from polypropylene or polyethylene, and the particle size of this sorbent bag is usually around 40 micrometer, whereas the frit size or pore size is round about 20 micrometer. This frits, the same frit is used at bottom and top, and frit should have good pore size. That is very much required. It should be able to hold the sorbent and should allow the mobile phase to pass through, allow the elution phase or solvent or your sample, if it's aqueous or whatever it is, they should be able to pass through this frit easily. These barrels can be fixed with these discs also. Other possible shapes are the first one. This is helpful if we want to have a multiple phases. If we have two of them, they can be interfixed with each other so that two phases or two different type of sorbents can be used. Why SPE is needed? Why? What is the need of solid phase extraction? Traditionally, the sample preparation consisted of sample dissolution, purification, and extraction that were carried out in liquid-liquid extractions. And if we talk about the disadvantages of liquid-liquid extraction, large volumes of organic solvents, cumbersome glasswares, and the cost also was high. Furthermore, liquid-liquid extraction often creates emulsions. When we are shaking the two immiscible liquids, the possibility of emulsion formation is also there, and this emulsion formation reduces or is a great hindrance for the extraction process. And also this solid phase can be easily liquid extraction cannot be easily automated. Another disadvantage with liquid extraction is it is an equilibrium process, meaning thereby that when we are having a liquid extraction, our analytes get partitioned between two different immiscible phases, and there is not 100% or transfer of analyte from one phase to another. So multiple extractions are required in order to completely extract the analyte from aqueous or any sample that we have. Whereas in solid phase extraction, this no doubt we can try for the multiple extractions, but even in the first extraction step, good amount of analyte is extracted. So these difficulties like incomplete phase separation, solvent consumption, solvent evaporation are easily overcome by solid phase extraction. Solid phase extraction was invented in mid-1970s and found use as an alternative approach to liquid-liquid extraction. And also we call it as digital step, because in case of solid phase extraction, two things are there. One, adsorption of analyte from the sample, and second is desorption. So sorption, desorption, or on off, or sometimes it is also called digital chromatography or digital extraction process. So different steps that are involved in solid phase extraction. The first step is the conditioning. This step is very important. Then is sample loading, washing, and elution. In case of conditioning step, which is the first step involved in the cartridge preparation, here solvent is passed through the sorbent bed to wet the packing material. So this particular conditioning step is required whenever we are to use the SP cartridges for sample preparation. We must take care that these cartridges are not dried before the sample loading. So conditioning step is important in this regard. So this is to wet the sorbent packing and to solvate the functional groups of the sorbent. Furthermore, air present in the column is to be removed and void spaces are filled with the solvents. Typically, the conditioning solvent is methanol, which is followed by water or aqueous buffer. The methanol followed by water or buffer activates the column in order for the sorption mechanism to work properly for aqueous samples. Care must be taken not to allow the bonded silica packing or polymeric sorbents to go dry. If the sorbent bed dries more than several minutes under vacuum, sorbent bed need to be reconditioned. So if we are having the successive extractions, if the sorbent bed get dries, conditioning is always important. If it is not reconditioned, the mechanism of sorption will not work effectively. And maybe to this conditioning step, cleanup step may also be added. During the condition, if necessary, the eluent solvent for this for the cleaning step, what we do is solvent is passed to the column after methanol and wetting step to remove any impurities that may be present in the packing material. This cleaning step would then be followed by methanol and aqueous again conditioning step, which prepares the column for sample additions. And if we look at the picture on the extreme right side, we will see that it is the C18 material And if this C18 material is treated with aqueous phase only, because this C18 or this is water or aqueous phase is highly polar and C8 is non-polar, and in presence of incompatible environment, this C18 may not be that much activated. So a solvent has the conditioning step requires the use of methanol so that this is activated. The next step is the sample loading. It's also called retention step, because in this particular step we are able we hold the analytes on this. So sample is passed from the top into the SP cartridge, and depending upon the type of sample and the concentration required, we can fix the sample size. It can range from 1 milliliter to 1 liter of sample, and this movement of sample from the top may be under gravity, pumping, or under vacuum, or by any other automated systems. It is important that the mechanism of retention holds the analytes on the column while the sample is being added. The mechanism of retention involves the various forces, which includes the van der Waals interactions, hydrophobic partitioning interaction or reverse phase. It can be, the retention can be through the hydrogen bonding. This retention can also be through the dipole-dipole interactions, size exclusion, cation or anion exchange. And during this retention step, analyte get concentrated on the sorbent bed. Some of the matrix components may also be retained, which is not a good sign, but they can be washed with the use of water or some other solvents. The matrix component, if they add to impurities, so because of whenever we are taking the environmental sample that contains many things, some analytes of our interest, some things that we really don't want. So when they are passing through, they get adsorbed with various under various mechanisms. The aqueous phase passes through it. The next step is the washing. Third step is the washing. Is to rinse the column of interferences and to retain the analyte. This rinse will remove the sample matrix from the interstitial spaces of the column while retaining the analytes. If the sample matrix is aqueous, the aqueous buffer or water organic solvent mixture may be used. If the sample is dissolved in organic solvents, the rinse solvent could be the same solvent. So after the washing step, which removes the interference, the next step is the elution. So in this step, the analyte is eluted from the sorbent with the use of appropriate solvent. your analytes need to have good solubility in that particular solvent, and also that solvent should be able to easily evaporate also, because that will even further concentrate the sample. So the next, what are the benefits of SP? The first that I told you is the sample enrichment. Other benefit that we have regarding the sample SPE is we get a very clear baseline. If we see the figure over here, we have a blank sample which is not made to pass through SP cartridges, and we have the spiked samples. We see that the baseline is not very clean. But if the sample is passed through the SPE, then the sample is quite clean of the interference and the baseline becomes clear. And because of this clean baselines, or because we get free from the contaminations of the sample, our actual peaks are easily seen. So we can compare the first and the second one. Here another chromatogram showing a non-SP sample and SP sample. And we see that the baseline cleaning is quite good in this case. Another application or another important use of the SPE is we can fractionate the samples by use of different polarities of the solvent. So polar compounds could be collected as separate fraction from non-polar compounds. If we look here, the fraction 1, the elution is done with water, and this will clear it of the polar analytes. The fraction 2nd, we have 8% of isopropyl alcohol. So another class of analytes will be eluted. If we increase the isopropyl content, the polarity of the solvent will decrease, and we will have another type of analytes which get eluted. And if we again change or increase the polarity, we will have non-polar analytes. Another important benefit of this particular SPE is, if this is particularly important when we talk about the use of mass spectrophotometer or GCMS as analysis technique. The upper output is signal of our compound of interest when injected in the saline. The lower trace shows significant reduction. In lower case, what happened is this is analytes in simple saline, but the lower one is the sample analyzed. First one analyzed when we are analyzing the direct human plasma. But in the first, this reduction in signals is because of the protein precipitation, because of the interference of the, or because of the matrix interference which cause the ion suppression, resulting in poor signal response. So we can compare these two. First, when only simple protein precipitation step is performed, SP is not done. In the second, when we perform the SP, analytes get removed, and those which were causing the ion suppression, they are not seen. This is how the SPE extraction system look like. Here we will see actual in the next lecture, where we will have the live demonstration of the working of SPE. There we will be dealing with more problems. Here we have glass chamber at the bottom, and on the top we can mount any number of SPE cartridges. And in order to enhance the speed of extraction, we can apply the vacuum. But in case the flow rate is okay, flow rate is very much important when we are talking about the extraction, that is step second, loading. Because if flow rate is very high, the analytes will not get retained on the sorbent bed. So we can fix the pressure as required by us. So drop-wise movement of the sample from the top to bottom is always preferred. And this drop-wise movement gives sufficient time for the analytes to get absorbed on the solvent bed. As regarding the milestones in development of solid phase extraction, earlier charcoals were being used, activated charcoals. Then in mid-70s, silica-based chemically bonded phases were, and even today, the silica-based chemically bonded phases are being used. In early 1980s, short pre-columns introduced for automated solid phase extraction were introduced. And particularly here in 92, solid phase micro extractions. Earlier they were silica based, then many other things were there, but more importantly, when we talk about the automation, this came with the solid phase micro extraction techniques and new sorbent materials like molecularly imprinted polymers introduced as the synthetic analogs for immunosorbents. So this is the timeline. And but now there are many more developments into solid phase extractions, which include the stir bar solid phase extraction, magnetic solid phase extractions. MOFs are being used as solid phase extraction materials. Fabric phases are being used as extraction materials. So there are lot of possibilities and novelties coming up in this new solid phase extraction materials. And also their use, not only the material, but how they are being used. They can be used by simply filling the cartridge, or they can be molded into tubes, or they can be coated inside the silica columns, or they can be coated on the silica surfaces on or inside. In case of solid phase micro extraction, we have coating on the silica syringes. These are different modifications of SPE. I told you that SPME is equilibrium extraction technique, whereas SPE is exhaustive extraction technique. Exhaustive extraction technique means it completely extracts the analytes from the sample, whereas when we talk about the SPME solid phase micro extractions, they do not completely extract, but they work by equilibrium extraction. So here the analytes are diffused toward the sorbent, stabilized on the substrate to establish equilibrium between the sorbent and the bulk medium. The analyte mass transfer towards the sorbent continues until the partitioning equilibrium is reached. I will talk about these different possibilities later in the lecture if time permits. But if we look into the, no doubt we have various other possibilities, but if we look into the actual use, or then SPE is most commonly used technique in the pharma industry. This will be followed by other dispersion or magnetic SPE or matrix solid phase dispersions. SPME also holds a good share of extraction material. Stir bar solid phase extraction is not very commonly used. Pipette tip solid phase extraction. In this case, a solvent is packed in a tip and to-fro motion of the syringe is done in order to extract the analytes from the sample. So this is also not very common. Now, if we look into the mechanism which are involved here, if we have aqueous sample, then a non-polar, if we have aqueous sample and non-polar analyte, then reverse phase chromatography is a good requirement, whereas if we have a polar analytes in aqueous sample, then reverse phase or carbon polymers can also be used. But if we have polar analytes present in the aqueous sample, then the use of ion exchange will be, ion exchange columns or ion exchange SPE cartridges will be required. And aqueous samples are most commonly, because whenever we are talking about the environmental analysis, it is only the aqueous samples. In case of non-aqueous samples, the decision of sorbents are somewhat reversed as compared to what we do it for the aqueous samples. Here the analyte that is polar and ionic. Polar and ionic. If our sample is polar and ionic, certainly we are going to use ionic ion exchange. And this is similar to the aqueous. In case of aqueous, we have the same thing. If analyte is polar but non-ionic, if analyte is polar but non-ionic, then we will be using normal phase sorption material. And if our analyte is non-polar, then we are going to use the reverse phase. Some alteration can also be there when based on the sample and sorbent materials. So new sorbent materials that are available, most common sorbent materials which are there for SP that I have already told also. They are silica based, they are most commonly used. When we see C18 or C8 or other, they are based on silica as a base material on which C18 or other chains are bonded. Polymeric materials are there. Magnetic materials can also be used. This magnetic material, basically the core shell in these cases is the ferrous based, and the analytes which we want to extract, they and the coating of these magnetic materials is done by the certain materials, the polymers, as well as on the top we have some ligands which can selectively extract the analytes of interest. Molecular imprinter polymers is another class. I will go into detail of that in later slides. Here, these polymers are made by imprinting the analytes of interest, metal organic frameworks, layered double hydroxides. They are also being used as solvent materials, fabric phase, graphene, and carbon nanotubes. In order to understand the sorption by SPE, it is important to understand the solid phase. So if you look into this slide, we see that C8 or C18 columns or C18 cartridges that we have, they have basically a silica backbone on which is bonded dimethyl octacylyl or 8 chain or 18 chain carbon molecules, and they basically make this silica surface hydrophobic. This is a brush-like appearance. It consists of reacting a silica surface with dimethyl octacylyl chlorosilane at elevated temperatures, which causes the bonding of dimethyl octacylyl group to the surface. As a result, the product is surface covered with diethyl octacylyl chains like the bristles of the brush. The term brush phase is sometimes also used for this. This monofunctional bonded phase was originally the most popular reversed phase solvents. And this brush having 8 or 4 or 18 carbon atoms attached, these phases are called C8, C4 or C18 cartridges. More recently, other bonded phases have been developed. We will see that C2 or phenyl groups or cyanide groups attached to this backbone. But if we look into the structure of this, the basic silica is polar in nature, which will not be good for the aqueous samples. So if our analytes are present in the aqueous sample, because this aqueous sample will deactivate the silica and it will no longer able to hold the analytes of interest. And these long organic groups, they make that surface hydrophobic, which can be used in the reverse phase manner. Still, these silanol groups which are present, they can cause the problem. So the concept of end capping, was developed by derivatizing the acidic hydroxyl groups on the surface of silica gel to prevent the unwanted interactions between the analytes and the silanols. End capping consists of, we see this figure at the bottom. Here, the OH groups of silanols, they are replaced. So now these are no longer available for hydrogen bonding. So end capping consists of reacting the derivatized silica gel with trimethyl chlorosilane reagent. This end capping process makes the overall surface of silica gel somewhat more hydrophobic, thereby creating more uniformly non-polar stationary phase. Another consideration which we talk about on the silica columns is hydroxyl groups which may be present as free silanols, vicinal silanols, or geminal silanols. And when they interact with the analytes, they interact in a different ways. Thus they may cause certain problems. Another thing that we need to consider here is we can see here the basic silica bed, and this water is retained or trapped in different forms. This water need to be removed. This water is retained in the void phases of the solvent, as well as adsorbed on the silica surface through hydrogen bonding and dipole-dipole interactions. These types of water molecules are removed by heating or under vacuum. The water in the smallest pores and water that is sorbed onto the silica surface will be highly trapped or bound to the silica. This tightly bound water will greatly affect the water that is present here. They greatly affects the recoveries from the bonded phases. And this is particularly important if the eluent is immiscible with water. This is another the common sorbents that we have. I have already told you, if we can class them into different categories, C8, C18, C2, cyclohexyl, phenyl groups bonded on the silica, or graphitized carbon. These all are hydrophobic in nature and form the reverse phase. And the next, that is normal phase. We have cyano group, amino groups attached, or diols, or simple silica gels, fluorosills, or alumina. These are polar in nature, and they are particularly important when we have polar analytes. For ion exchange, we have again amino groups. When we're talking about amino groups linkage, it is all it can be used as ion exchanger or as normal phase, but all depends upon the pH. At lower pH, this same amino group, they will be ionic in nature and they will act as an ion exchanger, whereas at high pH, this will be a normal phase material. When we talk about sulfonic acids and these strong acid groups, they remain invariant and always work as ion exchanges. Out of this, if we see the sulfonic acid, they act as a cation exchanger by the exchange of H positive ions with the cations in the analyte in the sample. Carboxylic acid bonded silica phases, they also work as a cation exchangers. Quaternary amines, they act as an ion exchangers. Amino groups attached, they also, if taken at a lower pH, they will act as an ion exchanger. These are the examples of silica materials, coatings. Now coming back to the materials which are packed in the sorbent, in the SPE cartridges. I have already discussed about the silica columns, silica packing. A disadvantage of the silica packing is that we don't have choice of the pH range. At low pH, this silica will dissolve or will cause a problem. So polymeric materials have wide range of applicability as regard to the sample treatment, because they are not much affected by the pH conditions. In this regard, I'll be talking about first material, that is molecularly imprinted polymers. This is area of my interest also. In this, molecular imprinting is a process where functional monomers and cross-linkers are co-polymerized in presence of the target analytes. So this is basically a process of co-polymerization. But this co-polymerization occurs with the template molecule, in presence of the template molecule. And once we have this template polymer formation with the template, next step is to remove this template. Next step is to remove the template. When we remove the template, the cavity, or when we remove the sample to be analyzed or analyte, your template is basically the analyte of our interest, which we want to analyze. And when we remove that analyte, a cavity is created, which works as a, hello? which works as a recognition site for the analytes of our interest. Main advantage of this MIP, that is molecularly imprinted polymers, is their high affinity and selectivity. When we talk about simple silica materials, there's a lack of selectivity. MIPs has higher physical strength because they are polymers, robustness, resistance at elevated temperature, high pH range, temperature withstanding is also quite high. Inertness against various chemicals compared to biological media such as proteins or nucleic acids. Their production cost is also low and their lifetime is quite good. So these things make the this enhances or this drivers to use the new materials for the sample for the SP procedures. So here we have prepared a molecular imprinted polymer for the extraction and analysis of carbamazepine and oxcarbamazepine in aqueous samples. Carbamazepine and oxcarbamazepines, these are anti-epileptic drugs and mood stabilizing drugs used primarily for the treatment of epilepsy and bipolar disorders. They are emerging pollutants in water due to their toxicity. When I'm using this word emerging pollutants, when I'm using this word emerging pollutants, these are new class of pollutants which were not earlier placed in the class of pollutants. So they have potential to work as a pollutant. The removal of CBZ, that is carbamazepine, by sewage treatment plants is often very low. So good quantity of that goes up in the surface water. And if it goes surface water, it has vision, dizziness, impaired task performance, or hypersensitivity. So this is how a schematic representation of how this is prepared. MA is methacrylic acid. Carbamazepine, that is analyte of our interest, and this monomer, carbamazepine and monomer, they are taken in ACN. 1 millimolar carbamazepine and 5 millimolars of methacrylic acid. These were dissolved in 30 ml of ACN in glass culture tube at room temperature. And the solution was ultrasonicated for 5 minutes to allow the homogeneous dispersion between the template and the monomer. Homogeneous dispersion. So this step is important. Otherwise, if we directly polymerize the polymer, then this particular analyte will not be trapped homogeneously in whole of the polymer matrix. So first, the monomer and CBZ are homogenized, and after that, the EZDMA, that is a cross-linker, and AIBN, which is a initiator, are added, and the polymerization process occurs. This occurs at 60 degrees for 16 hours, and we have a complete polymer. The next step is the removal of this analyte from the, because this analyte is bonded to the polymer matrix through hydrogen bonding and pi-pi interactions also. And this need to be removed, and this removal of template is done by succinylate apparatus using methanol methanol and acetic acid mixture, where they are added in 9:1 ratio. So we have a molecular imprinted polymer which have a site which is suitable for the extraction of CBZ. This is a SAM morphology. SAM, we show the morphology of imprinted polymers, and this suggests that they have a spherical shapes, and this spherical shape is good because they have a good surface area for the adsorptions, and this possesses good porosity and exposed surface area. This is this spherical shape is same for molecular imprinted polymer as well as non-imprinted polymers. This figure shows, because we have used this MIP for this figure contains the regeneration cycles which are possible. Regeneration cycle means how many times a particular material can be reused, and we have seen that this MIPs we were able to use multiple times. This particular thing is not possible or with the silica based materials. The recovery of CBZ indicate that, and this recovery is uniform throughout the end. Analysis after extraction of analytes using MIP was done with HPLC, and this table shows the parameters for the HPLC, where regression equation and correlation coefficients, they are very it is 0.997, which suggests that it is quite linear. Retention time for CVZ is 6 minutes, whereas for oxcarbazine is 4.7. Calibration, a limit of detection are quite good using using the MIPs, which is 0.488 for carbamazepine and 0.515 for oxcarbazepine. The leakage of, when we talk about this formation of polymer, one thing is very important, that when we are removing using a succinylate apparatus with any solvent, it may not be 100% removal of the analytes from the matrix. In order to have the, but the problem arises if those which are not, those analytes which are not retained, which are retained, if they leak in the further processes, that is a problem. But in our case, the leakage was not, leakage of analytes from the MIP was not found. For many cycles. The 10 cycles that we have performed. These are the morphine printed SP conditioning parameters. Sorbent that we are used is our own material, which is packed inside in between the two threads. And the conditioning solvent that we are using is water and methanol, 2 ml. Loading because we have sample in the water. So loading is done in aqua sample, 10 ml. And this aqua sample is spiked with carbamazepine, and the mixture of carbamazepine and oxcarbamazepine. HPLC conditions, C18 column is used. Mobile phase that is used is water, methanol, ACN in 40:30:30 ratio. Injection volume, this is standard, which is of the sample loop, 20 microliter. Flow rate is 1 ml per minute. The Dynex HPLC system is used, and wavelength selected is 250 nanometer. So the first figure 1 is when it is without SPE, and second is with SPE, the same sample after pre-concentration. This is a direct injection of the sample. This is injection after SPE. So we can see enhancement seen in the peaks. Also, if we look into the peaks, with carbamazepine is quite high. This particular behavior suggests that the MIP, that is molecular imprinted polymer, that we have prepared using CBZ as our template molecule, fits quite well for oxcarbamazepine, which is having a similar structure, but it is not able to retain the pyrimidones. So while the sample loading, these two carbamazepine and oxcarbamazepine are retained on the sample material, on SP material, whereas pyrimidone is not very much retained. Some is retained, but not very fully retained. So this suggests the selectivity of the MIP. Another type of materials that we have been using are porous molecularly imprinted polymers. Again, here the porous molecularly imprinted polymers were prepared, and the porous molecularly imprinted polymers, basically in this particular case, we have polystyrene core, which is coated with silica, and then on that we have the MIP. And this core of polystyrene is removed using THF, giving us a porous molecularly imprinted polymer. This porous will have a higher surface area, thus it will increase, or it will have more binding sites and increase the adsorption. How this is synthesized, I've been asked to be a little fast. So here the polystyrene spheres were prepared by the use of styrene, pyropylidene in triple distilled water at room temperature, and these were prepared with the reported procedure. So this was added initiator, which is potassium persulfate dissolved in 20 ml of triple distilled water. All conditions were done under the constant nitrogen temperature. Inert atmosphere is very much needed for this type of reactions. And the temperature required was 70 degrees for 12 hours. The resultant, these polystyrene spheres were suspended in the mother liquid. And after that, this suspended polystyrene was added the CTAB TOs, that is tetraethoxysilanes, coating of silica layer on polystyrene spheres. Here 1 gram of polystyrene spheres were added into the mixture containing 0.5 grams of CTAB and 20 ml of distilled water, 10 ml of ethanol, and 1 ml of ammonium hydroxide. TOs was added dropwise, and the mixture was stirred with the help of magnetic stirrer for 48 hours at room temperature. CTAB was removed using ammonium nitrate solution, and mesoporous silica was obtained by centrifugation and washed alternatively with the triple distilled water. And this prepared material, the polystyrene, was removed using THF washing. So what we get is the porous silica shells. Silica which is porous in nature. And on this silica bed, the polymerization was done. Here again the same procedure, we have carbamazepine, methacrylic acid, and ACN. That is allowed to stand, and to this is added PSS and EZDMA, which is giving us the imprinted polymers, porous imprinted polymers. And to make it molecularly imprinted polymers, this CBZ need to be removed, and this removal is done by the use of methanol acetonitrile in 9:1 ratio. These polystyrene spheres that were spherical in shape, and when we see the porous molecular imprinted polymers that were obtained, the SAM study shows that they have good pores which make them more good material for the as an adsorbent. Analytical performance using the HPLC we had good retention times, and limit of detection that was achieved was 0.082 nanograms per ml. The regression was linear. This is the important point here. When we are using the SPE, the optimization of SPE is required. The optimization parameters that we do in the SPE. We check the effect of eluent solvent. In this case, we tried ethanol, ACN, acetonitrile, methanol. A1 is methanol acetic acid mixture, A2 is your methanol acetic acid mixture, and A3 is methanol acetic acid mixture, which is 9:1. And we see that A3 gives the better elution. So this was selected for all the elution steps. Regeneration cycles for the prepared MIPs. This is also because it has to be checked over a period of time. Multiple uses are done. It has been found that even after 20 cycles, this was giving good results. Recovery was quite good. Other than the use of a type of the eluent solvent, we need to check it. What the minimum amount of solvent which can elute the best. We have seen that we tried it with 1 ml, 2 ml, 3 ml, 4 ml, and we found that with 1 ml, the results were quite good. With the use of 2 ml, slight addition was there, but very high recovery was achieved using 1 ml. The flow rate is also another important parameter. If flow rate is high, the recovery decreases. This is because when our flow rate is high, the analytes don't get much time to get adsorbed on the stationary phase. Adsorbent amount is also very important. Higher the adsorbent amount, more will be the recovery. But we cannot keep on adding higher amounts. We have to find a minimum amount which gives the best results. And we fix that adsorbent amount for our analysis. These are the chromatograms of carbamazepine in different samples. These are recoveries in different environmental samples. No doubt these are the spiked samples. We have applied this porous molecular imprinted polymers for the detection of diclofenac also. And in this case, our matrix was similar to what we have done initial, but the template was decided with diclofenac. Diclofenac is widely used for relief from pain, including rheumatoid arthritis, sports injuries. According to International Marketing Services, approximately 940 tons of diclofenac is consumed each year. So consumption is very high, and we have this high consumption. This is basically coming into the environmental samples also, into the dead animals also, which some reports were there that they have caused the reduction in the vulture population. So these were prepared. These are the SAM images of the porous molecular imprinted polymers. And here also we can see that these have the porosities, which is not there in the non-porous materials. This is polystyrene coated with silica. This is the porous silica. And when we talk about the porous silica, we see that the peaks which were absorption peaks which was seen here, they are not present in case of PSS. And also these peaks which were there in polystyrene, these are absent because polystyrene has been removed from the PS, from the porous silica by use of THF. In porous molecular imprinted polymers, we have the peaks which are associated with our cross linkers. Here again, we have tried to optimize the SP procedure by use of the adsorbent amounts and the eluents. And we have seen that A3, which is something missing here. Here also, methanol acetic acid is being used as eluting solvents. Adsorbent amount in milligrams, 40 milligram was sufficient. Reusable cycles, good number of reusable cycles are there. This can be reused. Elution volume. For this also, the 1 ml was quite sufficient amount of solvent for the elution. Flow rate was adjusted, and 1 ml per minute flow rate was quite good. Higher flow rate greatly reduces the extraction of analytes onto the solvent. And these elutions are done in methanol acetic acid 9:1 ratio. This is a table bar graph showing the comparison between C18 and porous molecular imprinted polymer for DCA, diclofenac, and we have seen that in various samples, the recovery that is achieved with C18 is quite low compared to that achieved using prepared material. So these are the environmental analysis results. Magnetic solid phase extraction is another technique. Here, the core shell of ferric, we get the magnetic materials, the core shell, and on that, the polymerization is done. On that, our material is coated. So these are functionalized cores. These are basically Fe3O4 nanoparticles and functionalized by use of various adsorbents with ligands. These ligands will have a selectivity for particular analytes, and if they are placed, if we have analyte which can bind to the coatings of the magnetic cores, coatings which are made on the magnetic cores, they will bind to these magnetic particles. They are functionalized magnetic sorbents. And if we place the magnet, it is concentrated at one site. So this is one of our publications which has been for the analysis of cadmium ions. Here we have prepared the magnetic core and coated that with the ligand of our interest, which was used for the extraction of cadmium ions from water samples. Another materials that is quite often used these days are layered double hydroxide for the adsorptions. Layered double hydroxides, they have a layered structure. And this layered structure contains metal ions in +2 and +3 oxidation states, and intercalation of carbonates or water in the basal spacing. And these carbonates or these anions can be ion exchanged with some other materials to make them slightly more hydrophobic, to be used as a reverse phase and to be used as ion exchanger for SP purposes. So we have tried the formation of these layer double hydroxides for alkyl phenols. Alkyl phenols are one of the well-known endocrine disruptors. And these alkyl phenols are easily transferred from bottles or packing materials into the edible products. And exposure to alkyl phenols are reported to cause various health concerns. So there was a need for to develop more materials which can selectively extract alkyl phenols for the analysis purpose. The LDH that's been prepared, this is a semi-maze, and we see that they have a layered structure. And this LDH was intercalated with dudecyl surfactant, and because of that intercalation, there's slight change in the surface morphology. There are cracks, but that also increases the surface area, but the basic structure of layered remains the same. These are HPLC consideration. These are analyzed using GCMS. Retention time was 5.94, 9.44, and 10.71 minutes. And base and peak was 77. These are the optimization parameters. Here again, different solvents were tested. Acetone was found to be better eluting solvent for this. Elution volume 1 ml was quite good. Flow rate 0.5 ml per minute, that was quite good for the elution. Adsorbent material 100 milligram was quite good for the higher amount. Higher amount is not decreases the extraction. Then pH range, we can see that it works well in pH range 3 to 10. So good range of pH for analysis. Salt addition, because when we add salt to the sample, that increases the analyte extractions, because they decrease the solubility of the analytes. But higher amount of salt, they will cause the problem, because water molecules, they will hydrolyze on that, and that will decrease the availability of water for the analytes. So minimum amount of salt that gives good recovery is taken. So these are basically all the SP parameters which need to be checked for the elution. These are GC parameters. This is intercalation of our surfactant into the layered structure, and we see that there is slight increase in the layer spacing, and the hydrophobic chains allow the hydrophobic interaction with the analytes of our interest. So normally if we use simple LDH, they will not be very efficient for the analysis of these organic analytes, but use of this integration make this slightly more hydrophobic, and so retention of analytes is done. Regeneration cycles show that they can be used quite for long cycles. This is the SPGCMS chromatograph recoveries. Other materials that are being used in literature are graphene and carbon nanotubes. They has been used as a solvent for speciation of chromium. If we look into chromium 3 and chromium 6, chromium 3 is needed, but whereas chromium 6 is toxic. Therefore, appropriate methods are required for their analysis. So graphene and carbon nanotubes have been quite well used in the literature for the extraction. So these are the new class of adsorption materials. Another important materials that are being used are MOFs. MO has a metal organic frameworks. They are three-dimensional network of metal organic framework which have porosity in between, which act as a good binding site for the analytes. So these are also quite good used for the solid phase extraction. Another materials which are common finding good use are supramolecular adsorbents. Here the supramolecules like cyclodextrin, calixarenes, crown ethers, they are immobilized on the silica bed or some other polymeric material, and they act as a good selective material for the extraction. Fabric phase is another material which is used for solid phase extraction. It integrates the benefits of sol gel coating technology and rich surface chemistry of cellulose. So on cellulose, sol gel coating is done that make it work as an adsorbent and used for the micro extraction of various analytes from the sample. This is a pictorial representation. We have a sample in that fabric phase is added. This fabric phase is coated with silica material by sol gel, and other modification are possible or derivation are possible on it to make it more selective. So analytes will get adsorbed. We can remove that fabric phase and put it into the eluting solvent. So all of the analyte get into this eluting solvent. We can take this sample and put it to the instrument for analysis. SVE finds good application as automation also. Normally, if I talk about this, between this port and this port, we have a sample loop in rheodine valve of injector port of HPLC. In case of sample loop, we can place coated materials, solid phase coated materials in form of silica coated silica tubes which are coated inside, and that can be used as a sorbent to make them inline sorbent material. So this is basically your load mode. What we do is we place the sample, pass through, it will get absorbed on the sample loop, because this particularly is specially coated inside, and rest of the sample will go to the waste. So this is basically automation, which is possible with SP, not with the liquid chromatography, and particularly with the polymeric phases. And once whole of sample has passed through this particular silica tube, which is coated with the material of our interest, analytes get absorbed, and next we can put it into the inject mode. The inject mode will bring our eluting solvent, which our running solvent, and will wash the sample, analytes, and pass it on to the column for the further detection. So in one step, automation can be achieved using HPLC. With this, I end my talk. If you have any queries, you can ask. Thanks for your kind attention. For all, my special thanks out to Irshad and Aman. A lot of work that has been shown here has been performed by them. And my special thanks are also to the HRDC and the GIAN team, Professor Gulshan Bansal, Professor Ashok Malik, for providing me a platform to share my work here with the audience. Thank you. Anji sir. Hello. Hello. Hello.
And you have tutorial now, na?
Anji sir.
Anji, Dr. Saab.
Anji sir. Tutorial nahi chalna sir.
Upon if there's any query regarding the theoretical concepts of this solid phase extraction, so the participants can ask, and otherwise we will now move to the next part where Dr. Hollock will take us to the laboratory, where we will see the demonstrations of working of solid phase micro extraction. That's right, Dr. Hollock?
Yes, it will be done, sir.
Okay.
But did they have the break or not?
Actually, break we cannot afford now because Dr. Bharti will be joining us at around 3:30.
Okay, so I have 45 minutes
we
for
have
that.
to wind up this session in another 45 to 50 minutes.
So I will do that.
Right? Because of the actually technical snags, now we had
It took a long time.
a lot of time.
Now it is working.
Thank God. So participants, so if there is no query, should we move forward to the next session where the demonstration will be there? Any response?
Yes, sir.
Okay, so fine. So there is a response from the participants. We can move it to the next session.
So before moving to the next section, I need to
tell something regarding that also? What we are going to perform. So tutorial in this, so am I audible now?
Yes, audible.
The slide is being shared.
Yes, it's visible.
Okay, sir. Okay, sir. So in this particular session, we will be performing solid phase extraction using LDH as our absorbing material, and the analysis will be performed using HPLC. The analytes that we are going to use are polyaromatic hydrocarbons, and if we look into the HPLC that we are going to use here, HPLC is simple chromatography technique where we have a solvent passing through the pump, injector where we inject the sample, it passes through the column, and which is analyzed. I think we must move to So I will be taking the polyaromatic hydrocarbons aqueous samples. Polyaromatic hydrocarbons are large category of persistent environmental contaminants comprised of 2 or more fused aromatic rings. Chief source of polyaromatic hydrocarbons in environment is attributed to anthropogenic activities such as motor vehicle exhaust, residential heating, industrial waste. You can see that there are large number of sources which are giving us polyaromatic hydrocarbons. Polyhydrocarbons are particularly problem because they are stable and cannot be easily degraded. They are significant environmental concern due to their bioaccumulations. So, keeping these things in mind, we have selected this. And because while performing or live demonstration, I will not be able to go for the optimizations because large number of parameters are required and that takes long time. So I will just show you some slides of the optimized parameters, and we will be using the optimized parameters for evolution only. Various solvents are selected. Out of that, elution with SP is best suited with hexane, and selection of appropriate eluent is very important because whatever we have done earlier, all steps, until unless it is eluted good, extraction or whole of the analysis will go waste. This eluent should have high affinity towards the target analytes. Various eluents such as were selected and recoveries of these are found in the health. If you look into the volume, volume 1 ml was quite good. Higher volume is not needed because most of the extraction is done with the first ml only. Flow rate that we are going to use is 0.5 ml per minute. Higher flow rates is will reduce the extraction. As far as adsorbent amount is concerned, 60 milligram of adsorbent will be taken, and I will show you how to
pack the SP cartridge. I will show you what are SP
वहाँ पे
cartridge.
दर्शन जी हमारी बात करवाना
You can pack it.
एफओ ऑफिस में ना दर्शन सिंह आशर जी हैं, उनसे मेरी बात करवाना.
And these are the pH conditions, and we see that recoveries are quite good at good range of pH, that is from pH 3 to 9. So that makes us use these new materials.
हाँ जी दर्शन जी. गुलशन बोल रहे हैं जी.
As far as salting out effect is concerned,
दर्शन जी अपनी वो फाइल ਜਿਹੜੀ ਸੀਗੀ ਪ੍ਰਵਾਨਗੀ ਵਾਲੀ ਉਹ ਗਿਆਨ ਪ੍ਰੋਜੈਕਟ ਚੋਂ ਮੈਕਬੁੱਕ ਖਰੀਦਣ ਵਾਲੀ.
salt addition,
कौन सी होनी है फाइल?
small amount is okay, but if we increase the salt to greater amount, that further decreases. So 5% salt was quite, NaCl was quite good for.
खत्म होने वाला है, फिर उन्होंने जल्दी सिरे लाइए आगे.
These are the HPLC chromatograms for the polycyclic aromatic hydrocarbons.
हाँ चलो ठीक है, कोई ना करेंगे. मैं फिर ਇਹਨਾਂ ਨੂੰ ਇਹ ਕਰ
The spiked samples.
नंबर लेना बेटा, मैंने नंबर.
This is
नंबर
the
नहीं है ना.
SEM image of the LDH, and these are layered structure. So I will just move to the lab now for the live demonstration. Do some charging. Good afternoon, dear participants. Here we are in the lab now. This is how the SPE cartridge look like. The simple syringe-like material made of polypropylene or ethylene or polythene plastic materials. And if we talk about the packing, these are the frets. Frets have in a pore size which is 20 micrometer. And this fret is packed inside, put inside. To go to the bottom. And on the top of it, we place the adsorbent, which is in this case LDH, that is layer double hydroxide. This LDH has already been prepared. And another fret is added at the top.
and care must be taken so that your material is packed to the best. You can press it from the top. Now SP cartridge is ready for the extraction of analytes from the sample. Next, that has got high resolution. Next, this is the SPE manifold. It is nothing but a glass chamber. On the top of it, we have the ferrules, which can hold or tightly hold the SP cartridges. So we have a possibility of having 1, 2, 3. In this particular case, 12 cartridges can be used simultaneously. So if we have large amount of samples or large number of samples, we can perform multiple extraction at the same time. At the bottom, we have a seal here so that there's a complete vacuum generated. This is nothing but a simple glass jar, quite strong enough to hold the large pressure, and pressure gauge is installed here. This particular, we have a base here which can be adjusted, and this base can be adjusted based on the sample requirement or sample amount. So my research student Aman will be performing the SPE process. She will be arranging, joining the pump with this and perform all the 4 steps of sample, your cartridge conditioning, loading, washing, and elution using this particular SPE manifold. In the meantime, because this will keep on going, in the meantime, we'll go into the lab where we will perform the HPLC of pH, polyaromatic hydrocarbons. So in order to have a comparison, we will perform without SP and with SP. The first injection will be done. Internet unstable. First injection into HPLC will be without SPE, so that process will go on, and we will make another injection after of the same sample, but after peak concentration, and we will see the comparison of these 2 HPLC chromatograms, and we will see the enhancement in the peaks. This enhancement will show that the analytes get loaded and analytes are
eluted means analytes are retained on the solid phase extraction cartridge, and they are well eluted using the elution solvent. So the first step, that is conditioning, is being done. Right. So she has switched on the pump. And what is your pass now? Conditioning, I told you, is with methanol and water. First, we pass the methanol. What is the reason for this? Means if we talk about the C18 columns, this is particularly important to condition it with methanol, because C18 columns has hydrophobic chains, brush-like appearance, which are hydrophobic in nature. And if they are very much close to, or they are with the aqueous phase, then because of the material which is unlike materials, one is aqueous and another is your hydrophobic, the SP materials becomes are not good for uses. So in order to activate them, it is important that we wash them with the organic solvent, and then with water, and then again with the organic solvent. This conditioning step is very needed to activate the SP cartridge. Yes. Now, so conditioning is done. Next step was washing. So what type of solvent you are using for the washing now? For washing, we're using simple distilled water. Because this washing step is needed, it washes away the matrix impurities other than those which are analytes which are retained on the solvent bed. Dr. Gulshan, am I audible to?
Yes, sir, you are audible.
Yes, it's audible, sir.
So, washing is done?
Yes.
Elution will be performing using the solvent which we have already optimized. And the optimized solvent is, which was that? Hexane, as I told you in the slide share. We elute it with the hexane. And care must be taken while elution. The drop or flow rate, if flow rate is very high, elution will not be very good, because like in case of your first step, that is loading. In that case also, if our flow rate is very high, then analytes will not get time to get adsorbed on the sorbent matrix. Similarly, if here the mobile phase or the eluent solvent is flowing at a very high rate, it will not get sufficient interaction with the analyte to remove them out. So in that case, we will be needing more of the solvent for the elution. So flow rate is important step both in case of loading and also in case of elution. In loading step case, it is important for proper retention. In case of elution, it is important for the eluent of the analytes. So sample is eluted. So what is the concentration of the sample?
So it is 0.1 ppb.
So-
After pre-concentration, it will become 1 ppb.
So 0.1 ppb sample was taken, and we will perform one injection of 0.1 ppb directly into the HPLC, and the second injection will be of 0.1 ppb, which is pre-concentrated and extracted using eluting solvent of 1 ml hexane as the eluting solvent. So because we are going to use these samples for HPLC, it is important that all these samples are a very good solution, homogeneous, and also they are degassed. Whenever we are using any solvent for HPLC, that has to be filtered. Number one, through the membrane filters of the size of 0.45 micron size, and they need to be sonicated so as to get degas them. So I am putting it in the sonicator, the sample. Switch on this.
Okay. Now it is audible. Oh. Hello. Oh. Sorry, there was noise of the sonicator which was creating problem. So this is the filtration assembly for the solvents connected to the pump, and care must be taken when we are not using it. We must open it. Otherwise, sometimes students they leave the tubes along with it, and this with this weight, because it's a glass assembly, it falls and breaks. So if you are not using it, you must remove it always. All parts I have separated. Now sample is sonicated. We switched off the sonicator. Now we'll moving to, because for HPLC we are using triply distilled water. This is the setup which is used for triple distillation. In the first chamber we added KMnO4, some beads to avoid the bumping of the solvent. So it is simple distillation where water get evaporated, condensed, comes to the second stage, then to the third stage, and that is used for the HPLC purpose. So now I am moving to the HPLC lab, where we have both the samples. One before pre-concentration and after pre-concentration. This is the HPLC system that we have. It's from Waters binary system. This is the column, C18 column we are going to use here. As we have changed the column, so parameters that we have, optimization parameters for HPLC that I have just shown you may not work, so we will check it out. These are the binary. So we have two pumps. The detector is PDA detector. I'm not going into the working of HPLC because that you have already seen with as previous HPLC lectures. And if you have any query regarding the HPLC user, you can always ask me. So the Breeze software is being used for the analysis. The HPLC column for HPLC injector is very specially designed injector. I will just share the screen. इंजेक्शन कर रहे हैं? बंद कर देना. So I will explain the SP injector later on. Meantime, we can have the injection of HPLC. So HPLC injection will be done in the injection port, which is a rheodine valve, and it is very specially designed rheodine valve, because to have a very fixed volume of the sample to be loaded, because that is very much important for the analytical purposes. If we load extra sample, it will come out. So Ms. Aman will perform the injection and share you with the various steps. In order to use HPLC, we have to first set the program. Program means what type of the flow rate is needed, pump pressure, because sometimes the pump pressure exceeds to a certain limit. So we have to set that red lines that beyond this pressure, it should not be accepted. Flow rate is important. Also, we need to see if it's a binary system, what ratio of the solvent is to be taken from which container. So that is also set in the program. As a detector parameter, I have to check out if it is a UV detector, what are the wavelengths at which my sample absorbs. So that is also, the wavelength setting is also done in the program. So we have got the injection done, and this was done using the syringe, the glass syringe. Which is whatever amount of sample we can inject. But the thing is, HPLC injector port, which is specially designed, it will take up that volume only, rest excess volume will come out. So what are the HPLC conditions you said?
So it is 70, 30, 70 is methanol and 30 is water.
70?
70-30 ratio. Points mobile is 70 is methanol and 30 is water.
Okay,
0.70, 0.30.
So methanol water ratio is set as a optimized parameter. 70 water, 30, 70 methanol, 30 water. These are the optimized parameters. And what is the flow rate that you said?
It's 1 ml per minute.
Flow rate is the rate at which the solvent comes from the column. Column. So that is set as at 1 ml per. So by the time this is running, run is of 20 minutes, and whole of the signal is being captured in HPLC
So these are the columns for HPLC. These columns, inside they have the packing material, and these packing materials on both sides are capped with frits. These frits are specially designed because we have the packing material, maybe C18 or any material. The HPLC pressure is very high, and that this whole of the pressure comes on these frits because they have the role of holding the stationary phase and allow the mobile phase to pass through it. So frits are porous but have a very high strength which can hold that high pressure. If frits are not good, then process of leakage of the packing material of the column can be there. These are the reservoirs for HPLC. Reservoirs means from where the solvent, the mobile phase is kept. And these are coming from two different tubes. These solvents are already degassed and filtered. They enter into two pumps, and then mixing is get mixed, and the ratio in which they enter into the pumps is set by the program. So in this particular high pressure mixing is there. We can have single pump system also. In those mixing is low pressure mixing, where mixing is done first of the two solvents, and then they move into the column, move into the pump, from where they are pumped into the column. It's low pressure mixing and high pressure mixing. So here solvent directly entering the pump, both pumps, and from there they enter into the mixing chamber, and then they are mixed. Now I will be showing you. So 20-minute run, so we have to wait for that. So if you have any query regarding HPLC, you can ask me. If anybody having any query regarding HPLC, yes.
Good afternoon, sir.
Yes.
Sir, I'm working on a drug which contain no chromophoric group. So I'm using some derivatizing agent for making some complex so that it will show some peaks on HPLC. But sir,
Okay.
the problem is up till now, whatever the work done, it was done on fluorescence detector.
Okay.
And I'm developing a method on PDA detector.
Okay.
So what are the steps I have to take? Because sir, while I'm using PDA detector over fluorescence detector, so you know, the concentration I'm using now, it shows it's not showing proper peak.
Okay. With
So,
PDA?
sorry sir.
With PDA detector, it is not showing proper peak. Proper peak.
Yes sir. Yes sir.
We need to change the derivatizing agents.
Yes sir.
Because it should have good absorbance.
Sorry sir.
It should have good absorbance because PDA detector is basically UV based.
Yes sir.
So if unless it is going good absorption, it will not be detected in the PDA detector. So first look for literature for derivatization where it can be used with the UV.
Okay, sir.
Once it is used for UV, you have a peak on UV, it will be seen the PDA also.
Sir, I will tell you, I'm using the drug Agmatin.
You can
Hello.
personally call me, I will check out.
Yeah, okay, sir.
Actually, just I want to add something to this. See, you said that your method for that is reported with the fluorescent detector, right?
Yes, sir.
So if it is reported for fluorescent, then it means by default it is absorbing in the range of 200 to 400 nanometer. Only
Yes.
shows you absorbance. Okay.
Yes, sir.
So it is already UV absorbing. If you are not getting a good peak shape, so it is not related to detector, it is related to the chromatography condition. Right.
Okay.
So to get a good peak shape, you have to optimize the mobile phase pH or the stationary phase. This is what we talked about last day about this chemical factors which are guiding the selectivity. Okay. One
sir.
thing is it may be possible that you may not have access to fluorescence detector, but you can do with PDA, but PDA has its own limitation. PDA is, you see, it is a good for qualitative work, but when it comes to quantitative work, PDA has a lesser sensitivity in comparison to the normal UV visible detector for HPLC studies.
Okay, sir.
Right? So if you have UV detector, so it is a better you go for the UV detector only, not PDA detector for quantitative purpose.
Okay, sir.
And if you want to increase the response, right? If you want to increase the sensitivity, then again, as Dr. Jitendra said, you may have to change the derivatizing material so that its absorptivity can be increased.
Yes, sir. Sir, can you please tell me what concentration I have to choose? Because whatever the research paper I am referring, they had used a 1:1 ratio, the drug and the derivatizing agent. But while I'm using it on HPLC, it show the peak was not proper shaping, and it's showing too much concentration of derivatizing agent. I had used some concentration on UV, but it's also do not show the proper result. So can you please, sir, tell me that which concentration I have to use? Yeah, by which method or technique it will confirm proper complexation was done?
That you have to optimize actually. Let's say
Optimization are required. Means
you can use
sometimes what we do before the column, because normally when you're taking simple UV, it is a one process, and using HPLC is another process. In HPLC, it is passing through a column which is having a stationary phase adsorbents. So how your derivatized system works with the sorbents, whether it is stable with that or not. It may happen because optimizations are required. Nobody can tell in the one go unless you see the system.
Actually, you have to optimize the things, right? Like you can take multiple proportions, like a 1:1, 1:2, 1:1.5, right?
Yes, sir.
Let's say you have taken one proportion of your drug, so you can take a equal proportion of your complexing agent and a derivatizing agent, then 1.5 times or 2 times, and then you can analyze it with the help of HPLC and see where you are getting the maximum derivatization, right? So this way only we have to optimize.
Sir, is there any alternative method by which we come to know that whatever the complexes form is that are compatible with each other, like derivatizing agent and its with drug? Is there any method?
No, that must be reported in the literature. Only on the basis of literature you are using it, no?
Yes, sir.
So if it is reported in the literature, it means they are compatible. And only point is you have to find out the correct conditions. So usually whatever the conditions are reported in literature, sometimes they are not, if they are just chemical conditions, they need a slight variation in the amount, in the time, right? So that you can reproduce it. So that needs optimization. Okay. Anyway, whatever your issue is, we can discuss more upon it. After the session, we can have a discussion on it, right?
Yes, sir.
But we will love to definitely solve your problem.
Yes. Thank you, sir.
Okay. Most welcome.
Hello.
See, Dr.
Hello.
Malik. See, Dr.
Yes, sir.
Can I know your name, please? Can you tell your problem to us? And because it is not necessary that complexation is reported in the literature, you can study it directly also. First, you take the absorption spectra of the drug and the complexing agent. You optimize the conditions, and under the optimized conditions, if your is working state, first condition for HPLC is your complex form must be stable in the mobile phase. If it is undergoing decomposition, then you cannot do anything.
Okay, sir.
Right. Okay, Dr. Alok, please continue.
Okay. I have shared one slide.
Dr. Alok, I just one submission that
Yes.
Dr. Bharti is waiting for his turn. So we have to wind up this session in a few minutes, right?
No problem, sir. My HPLC run is going to complete
Yeah.
in 2-3 minutes.
Okay. That's fine. That's fine. Thank you.
Narrative I will not able to, because that will take even 25 more minutes.
Okay.
So one simple run I have shown.
Yeah, sure.
So this I have shared one slide about the HPLC rheodyne valve. It has got two positions. One is the load position, another is the inject position. Between 1 and 4th port is a sample loop, and between 2 and 3 is your pump inlet and pump out, inlet of pump and column out. So when in the load position, what happens is we are injecting the sample, it gets into the sample loop, excess of the sample comes out. So excess loading will not have any effect, excess loaded will come out. And another thing is we can automate this also. We can have a good amount of polymeric material inside, which can act as a sorbent for inline solid phase extractions. So in load position, what happens is the tubing which was coming from the pump that comes in loop with the sample loop, and hence sample get removed from the sample loop and is injected into the column. So this has to be there because HPLC is working at a very high pressure. Simple injection will not work. This is very specially designed, which can change from one side to another at such a, and withstand that high pressure also. So now I'm again moving to the, okay. So this is HPLC run, which is before pre-concentration, where peaks are not seen properly. Some
And next run is of SPE pre-concentrated sample. We are not going to perform it now, but we have one result which was from the previous. We'll show that result. These are same polyaromatic hydrocarbons which were peaks can be seen after pre-concentration. They were in very low concentration before pre-concentration. If we perform a direct loop injection, but these show good peaks when injected after pre-concentration using SPE. So with this, I end. If anybody having any question, he or she may ask.
So if there is any query from any participant, please feel free to ask.
We have not performed the second injection because that will take long time again. The time doesn't permit us.
Yeah, actually, otherwise, Dr. Heminder Bharti's talk will be over by 5:30. So I think by that time your lab might be closed, right?
Okay.
Otherwise, we could have shown that thing after Bharti's session.
But these are runs that we performed 2 days back only. So these are equally good. Even after 5, you may see the similar results.
Okay, okay. That's great. That's great.
Wouldn't want to see live here in the lab, he or she is always welcome.
Definitely. Definitely, the participants are most welcome to see the facilities, to use the facilities which are available here. And this is the purpose of this course, so that we can be aware of the new things and we can see the facilities, so we can share the facilities for the better guidance, better results. Anyway, so if there is a no query from any participant, then I would love to thank Dr. Jatinder. This was the lab exposure, this session. This was a marvelous one, Dr. Jatinder. You have done a lot of efforts in making the things live. We wanted to do the things offline, but you made it offline in online mode. So that is very good of you, and your efforts are really visible. Thank you so much.
Thank you very much, sir.
So much.
It's a pleasure coming here.
Okay. So, guys, now we'll be starting our next session. Dr. Heminder Bharti. He will be joining in 1 or 2 minutes. On this topic of environment contaminants with our participants. Over to Dr. Heminder Bharti.
Yeah, thank you, Professor Gulshan. So it started in the morning. So fortunately, it has gone up to the evening. So I hope it is fine now. Hello.
Yes, it is perfect now.
Okay. So as I started in the morning, the topic, in fact, is the contamination of aquatic ecosystems, concerns, threat, and mitigation. So as I said in the morning as well, we are passing through a phase when there is a lot of environmental degradation, then ecosystem degradation, and so many other issues which we are currently facing, like at present climate change. So I will address all these issues with respect to aquatic ecosystems, that what are the concerns, what is going on at present, and what are the threats and how they should be mitigated, and what is the challenges which we are facing right now. So as far as the structure of my talk is concerned, first to build up the story, That is, what are aquatic ecosystems and what is their importance, why they are required, because until or unless we do not know the importance of aquatic ecosystems, there's nothing we can think of conserving or think of restoration or reversal. So for that very purpose, we need to think in terms of that what are aquatic ecosystems and what are their types. Then contamination, of course, that what kind of contaminants are there in those ecosystems and what kind of challenges or they are facing, the concerns, then threats, then mitigation, as I said earlier, and the challenges. So this particular epoch, although this is not official sort of classification of geological time scale, this particular epoch is at present known as Anthropocene. So it is an unofficial unit of geological time, which has been uh, considered as the one which has been heavily influenced or impacted by human species. So this is considered as the most recent period in Earth's history, as human activity have started a significant impact on planet's climate and uh, ecosystems. So this particular term is quite catchy nowadays. Instead of saying Holocene, we use the term Anthropocene because it is heavily influenced by human species. And now we know this was the report which was released last year, the Intergovernment Panel of Climate Change in 2021. And at that point of time, they have very clearly stated that humans have influenced or warmed the atmosphere, ocean, as well as land to such an extent that we are feeling the heat, we are feeling on the influences, we are feeling the impact on species as well as other organisms, and including human beings. So until or unless human species is such a selfish species, until or unless it feels or it feels the challenges or there is some sort of impact on this species, we never bother for anything. So we have come to a situation where there's a lot of influence on our species, our survival is threatened, our ecosystems are being destroyed. So that's why now we think in terms of these things. Now this is the report which was released last week, and it has some startling observations. That is the sixth assessment report which IPCC released on 28th February 2022. That was on impacts, adaptation, and vulnerability. That what are the impacts and what in terms of adaptation and how vulnerable are we or the other organisms. So at one point of time, we thought that we would be able to control the temperature below 1.5 degrees Celsius, but now at present, as the 175 nations, they collaborated together with about 300 scientists. And when the result came out, at present, we stand at 1.1 degree. So it is impossible that we can curtail this to below 1.5 as present or as the situation stands now. And given this rise in temperature, that is 1.1 degree at the moment. So the situation is that 1 billion are exposed to heat waves every 5 years. So if the situation goes like this or the current circumstances as they unveil or unfold with time, 1 billion will be exposed to heat waves every 5 years. And global sea levels would rise by 1.5 feet. So, and then threatening millions of species in coming years. Earlier, when this kind of thought or this kind of generalization or this kind of data was shared, when Rajendra Pachauri was heading this IPCC, people used to think that this is nonsense, nothing is going to happen, there's no rise in temperature, this is all proclamations which have no basis. But now it is very clear that this is on the agenda of UN, on the agenda of IUCN and other agencies or other persons who are thinking in terms of survival of other species as well as human species, that this is going to happen. And if it goes to 2 degree, then 5 billion will be exposed to heat waves every year. And global sea levels would rise by about 2.8 feet. So this is the kind of situation which we are facing now, and whether they are aquatic ecosystems or they are other ecosystems, everything is going to be impacted by this kind of change. So what is the importance of aquatic ecosystems? So as we say that aquatic ecosystems, basically they comprise of many different kind of ecosystems, and they include wetlands. Then we have lakes, which are vital for life on Earth because they provide us a range of services as well as the benefits. Then there are issues with stakeholders and others as in terms of, because everything is interlinked between sustainable development goals, which are set by United Nations. At present, they have 17 sustainable development goals. We call them SDG. And there is a sort of interlinkage between these goals of United Nations. So the purpose is to strengthen as well as to trade off relationship. So whenever we talk in terms of stakeholders, or whenever we talk in terms of empowering the public or the indigenous people, the problem is that there is an issue between trade-off and what we are getting out of it. That is a trade-off between benefits as well as what is being lost in that case. So it says that the sustainable development goal, it says that it ensures availability and sustainable management of water as well as sanitation for all. So as I said earlier, there are 17 SDGs which have been mentioned by United Nations. So out of these few, they deal with clean water and sanitation as well as the aquatic ecosystem. So that number 6 deals with this, that is availability and sustainability as far as management of water is concerned, as well as sanitation is concerned. So about 3 billion people depend upon marine and coastal biodiversity for their livelihood. But at present, what we see is 30% of world's fish stock, they have been overexploited and they are reaching below the level at which they can be produced. So we are not able, at present, we are not able to match the demand. And oceans also absorb 30% of the carbon dioxide, which we produce. And we are seeing only 26% rise in ocean acidification since the beginning of industrial revolution. So these are alarming facts which have been raised by United Nations, by scientists, by IPCC. So whenever we talk in terms of sustainability or whenever we talk in terms of conservation, we have the empirical data, we have the analysis with us, but the problem is that these things are not interlinked. We do not plan in a way that this is going to happen, how we need to curtail for the coming years, or how we need to curtail these things for in such a way that they are useful or sustainable for the posterity or for the next generations. So that is the kind of problem which management as well as sustainability is concerned. It's a sort of gaming cube that we start forming one side of it or start shaping one side of it, and in the meantime the other side is distorted. So these kind of situations which we are facing, and I think day before yesterday or even yesterday I was reading on a paper on Twitter, and I found that plastic pollution is the major issue which is being dumped into oceans, and those microplastic fragments, they are creating a lot of problem for humans as well as the species which are present inside oceans. So this is something alarming which we are facing right now. So as far as these marine ecosystems are concerned, they form 70% of our surface, and some of the data says that it could be 80% also. And as you know, they comprise of oceans, estuaries, and coral reefs. And these coral reefs and estuaries, basically, they are the most productive ecosystems, in the sense that when we say tropical ecosystems or tropical forests, they are the most productive, but estuaries and coral reefs, even they are more productive than that, as we have C3, C4 plants. And generally we see that C4 plants, they fix more carbon dioxide as compared to C3 plants. But these two ecosystems, coral reefs and estuaries, they are more productive as compared to oceans. Then in case of fresh water, although that is 1% of Earth's surface, but they cover 10% of the species which inhabit Earth right now. And they comprise of these three: lotic ecosystem, lentic ecosystem, then wetland ecosystem. So all these ecosystems, they play a major role in our survival. They provide us clean water, then sanitation and other sustainable services, which are being overlooked when we contaminate or when we pollute these ecosystems. So this is a sort of representation which has been nicely done by one of the agencies which deals with climate change. And you can see over here that whenever we talk, we generally say tropical forest, temperate forest, then we have savanna, then tropical land. In terms of carbon dioxide absorption and in terms of carbon sink, the carbon sequestration, that how much carbon these ecosystems can absorb. And over here, it is very clear that the wetlands, they absorb 40% of the species, they live in these wetlands, and this is the carbon sequestration which is contributed by wetlands. That is the highest amount because they play a critical role in regulating the global climate, and they store it as natural carbon sinks. So this is a kind of situation, this is a kind of you can say mitigation, or this is a kind of challenges which we are facing current now, as our wetlands are depleting and they are the major source of carbon sink. So what is aquatic pollution? Since when I was preparing this lecture and Professor Gulshan informed me that this is related with aquatic ecosystem, so I thought of this topic because until or unless we realize that what are aquatic ecosystems and what kind of aquatic pollution is there, the basic knowledge which we should have before we proceed for any kind of sustainable or conservation activity. So this was the very purpose of designing this, because as we say, as I said, the aquatic ecosystems are carbon sinks, but nowadays what has happened over time, they are the sinks of anthropogenic contamination. Then we have industrial wastes, leaks, chemicals, and other solid pollutants. And how this has happened over time, because there are numerous anthropogenic activities, and all those anthropogenic activities, they are created by human species, because human species or human beings, they consider that they are the most advanced creatures on this earth and they can do anything whatever they want to do. So that kind of selfish attitude has led to discharge of various pollutants into aquatic ecosystems, aquatic environments, and that is polluting the environment, damaging the quality of environment, and thus rendering those water bodies in opposite. So this massive quantity of agrochemicals, organic matter, then we have drug residues, then sediments, saline drainage from agricultural lands are discharged into water bodies. So it has become hazardous for aquatic environment as well as for human health and all those productive activities. So that is basically pollution is man's greatest crime, which human species has committed against itself. So contamination of aquatic ecosystems by anthropogenic activities, in the sense that what are the factors and what is the current situation. Deforestation is considered one of the major factors. Then we have construction of canals, dams, roads, filling and construction of canals, dams, roads, and bridges. Then we have agricultural activity, that is the impact of green revolution, which we started in 50s. We thought that we can increase the yield, but in the meantime, the problem was that with increase in yield, there's a trade-off associated with that. At that point of time, we focused on one thing, that in order to avoid hunger or to curtail hunger, we would increase the yield or we will increase the output. But the result is that indiscriminate use of fertilizers and other chemicals has led to aquatic pollutions, and automatically the soil is also drained of minerals. So this kind of situation, this is one example, one of the very good examples of human thinking, that at that point of time we thought of green revolution, but we never thought of the offshoot or the problems or the impacts associated with this kind of thinking. So that was the product of some scientists and they got the Nobel Prize also. But the offshoot is still going on with the human species, which we are still facing. So as I said earlier, the issue is that when we start some activity, we do not think in terms of sustainable or the offshoots associated with that, whether it is what is going to happen after 20 years or what is going to happen after 30 years. So this kind of management is lacking altogether in scientific arena, in scientists, in agencies, and different kind of stakeholders. Then we have industrial and domestic activities. So as the data says, that is of 2017, that United States is the major cause of pollution in rivers and streams, and is agriculture, as well as 30% of the water bodies under agriculture pressure in European Union. And in China, agriculture is predominantly responsible for surface water pollution, and that leads to groundwater pollution. So this is a sort of alarming data which is already with us due to anthropogenic activities, because at some point of time we are responsible for these kind of activities. So what are different kinds of categories of water pollution? So I know most of you might have read in textbooks or in your degree courses or postgraduate courses. So that is not my concern at the moment. My concern or perspective is entirely different. Just to give you a fair idea that what categories of water pollution are they and what kind of destruction are they carrying for us. So maybe groundwater, then we have surface water, then we have ocean water, then these different categories are there. And interestingly, the British poet W.H. Auden once noted that thousands has lived without love, not one without water. So yet, while we all know water is crucial for life, we trash it anyway. So the groundwater, because when as far as groundwater is concerned, it gets polluted when contaminates from pesticides and fertilizers. They are leased from landfills, septic systems, and make their way into aquifer, rendering it unsafe for human use. So this is one point. And when we think that we can purify that groundwater of contaminants, it is almost nearly impossible, as well as this is a costly kind of situation which is difficult to get on. And once polluted, an aquifer may be unusable for decades or even for thousands of years. So this is the impact which human civilization of human species has created on groundwater, taking it for granted that we can do anything, whatever we want to do. Then second is surface water. So as far as surface water is concerned, this is again a sort of report which is there. Half of our rivers as well as streams and more than 1/3 of our lakes are polluted, and they are unfit for swimming, fishing, and drinking. And nutrient pollution, you might have already had the lectures on this, like nitrates, phosphates. They had the leading type of contamination in these freshwater sources. And as we know that when plants and animals or other organisms which are present in these aquatic ecosystems, they need nutrients to grow. And when they need nutrients to grow, obviously these kind of nitrates and phosphates, they create another type of problem. I'll come to that later on. And municipal and industrial waste that discharges or contribute their fair share of toxins as well. So industry, individuals, then we have municipal corporations as well as human beings all contribute to pollution or contamination of surface water. And then we have ocean water such as chemicals, nutrients, heavy metals. They are there, which are carried from factories, cities, by streams and rivers into our bays, estuaries, and eventually they reach up to sea. So this is a kind of debris which is being created inside ocean, and including plastic. Because as I said earlier, I read a report day before yesterday that 175 nations, they have signed to created a situation. Now we are wasting a lot of money, energy, as well as thought process to curtail it just to make it fit for our future generations and for ourselves. So somewhere down the line, our planning is faulty because we do not know what we are going to do now and how it will impact our species for next 40 or 50 years. So then our seas are sometimes spoiled by oil spills, leaks. They are also there because they are also creating a lot of problem. Then man-made carbon emissions. So these kind of contaminants as well as emissions, they are. So what kind of point sources of pollution? So this is when contamination that originates from a single source. We call it as a point source pollution. And then examples include wastewater, that is effluent, which is discharged legally or illegally by a manufacturer, oil refinery, or wastewater treatment facility, as well as contamination from leaking septic systems, chemical, oil spills, and of course, in the Indian scenario, illegal dumping. So this kind of illegal dumping without any legislation, without any foresight, without any thinking, that also creates a lot of problem. And although we have established certain limits that what should be discharged by a facility directly into body of water, again the problem is that when we think in terms of this, that this amount has to be discharged, obviously this is for one body or for one facility. But if many such industries or other units are placed there, and then they combine together, we have an accumulative effect on the water body. So although it is called a point source, but at that point of time, when many discharge together, it becomes a sort of diffused source. So if it is at one place, for example, if this situation happens at one place, it can affect miles of waterways and oceans. Like recently, the Himachal government at the border of Punjab and Himachal, they have opened lot of industrial ways at that border, and all those, they discharge their effluents into Beas River, and eventually all those kind of effluents, they are polluting over there, but now they are running into the water body, and eventually they're going up to whole of Punjab and creating lot of problems. Then non-point source. Non-point is right, as I said, from diffuse sources. They may be agricultural, they may be stormwater runoff or debris blown into waterways from land. So this is also leading cause of water pollution, but it is difficult to regulate because since no single identifiable culprit is there. If you happen to travel to Himachal Pradesh, like this is a very crude example, then I won't shy of sharing it, that for example, during summer, all of us, they move to hill stations most of the time. And what we do over there, we carry plastic bottles, we carry beer bottles. And then if we go to hill stations, whether it is Himachal or Tamil Nadu, whether we are going to Shimla, Manali or Uti, we throw all that kind of garbage onto the land. And ultimately, if some river is nearby, it ends up there. So nobody is going to control us, and eventually what would happen, all that kind of garbage will end up inside the water body, and it would reach somewhere down the line up to the sea. So this is then transboundary that water pollution can't be contained by a line or map. It is resulting from contamination carried by one country and spilling into waters of another. So this is wherever we share our rivers, wherever we share water bodies, this kind of situation or these kind of problems always arise. So we need global awareness in that sense, that every country or every political arena should be aware of the fact that humans, they live in all countries and they survive on these kind of aquatic ecosystems. So contamination may result from oil spill, slow downriver creep of industrial, agricultural or municipal discharge. So transboundary or political regions, that is there, for example, Bangladesh and India. So this is a sort of depiction on the slide that what are the sources of contamination, that is agrochemicals, what is there in sewage, then we have heavy metals, and then there is another ecological issue, that is eutrophication. Then we have plastics as well as microplastics. Then we have oil spills. Then we have aquaculture, because at one point of time we thought that aquaculture is a productive industry. But over the time now we are realizing that aquaculture, instead of a productive industry, it is also a source of water pollution. So as far as agrochemicals are concerned, we have a data from Punjab from our students, and what we found is that there's a lot of pollution in water bodies as well as in the land also. So we have data, there is a canal which flows nearby Sarhand, and we found that when we have got the soil analyzed from the nearby area which is close to the canal, we found a lot of heavy metal contamination is there. So, and that contamination is basically restricted to that area or that soil which is close to the canal, and beyond that, that kind of heavy metal contamination or heavy metal accumulation is missing. The reason being because during rainy season, whenever the water flows up to the brim or just moves out of the canal to the nearby soil, those heavy metals or those kind of contaminants, they get accumulated in the soil nearby. So that kind of problem is also there, because if we are discharging lot of effluents into the canals, or we are thinking it will run away, that won't happen, because at some point of time, the soil nearby or the ecosystem which is nearby will also be impacted by that kind of water body. So this is another kind of example which is there. And as we know that the population is growing at an exponential phase, and there is increase in food demand. As I said earlier, there's green revolution, expansion of agriculture. And then we have unsustainable use of agrochemicals to increase the production, which basically contributes to pollution in aquatic ecosystems. Because whenever we have unsustainable use of agrochemicals, then the issue is that most of the time we do not know what quantity should be used, what kind of chemicals should be used, what kind of pesticides should be used, what kind of insecticides should be used, and how they are going to impact the human species as well as. Now, a simple example that at our homes we use this mosquito repellent. So that particular chemical we are using, and then there's one point of time. On other hand, the mosquitoes or other insects, they are having genetic resistance to that kind of dose. Then we go to the market and we look for another solution. Then the company comes up with double dose. Now, why that double dose is required? Because there is a genetic resistance in insects and other organisms, because because evolution or natural selection works in a sense that some of them survive, and eventually what happens with passage of time, that we have that breed or we have that kind of population which is not acted upon by that kind of dose. So at that point of time we need double dose, and maybe after 3-4 years we may have triple dose. So this way we will be increasing the dose, but at no point of time we will able to curtail that organism or that kind of insect from spreading the diseases, because our planning, our management is purely wrong. It comes to my mind one of the examples from Bombay. As you know that Dharavi in Bombay, it controls whole of the economy of Bombay. So in Dharavi, it is a sort of slum area, and what happened once that most of the population, due to slum and humidity, they were affected by Mycobacterium tuberculosis. So that is tuberculosis, and eventually when the disease was in full bloom, they ended up in the hospital, and doctors prescribed that this sort of antibiotic should be, for example, should be taken for 3 months. And when it was for 3 months, after 3 months, when there is no fever, there are no visible symptoms, they stop taking medicine and stop visiting the hospital. And in the meantime, the bacteria, it became resistant. Those populations, they survived due to those selection pressures, and eventually that population increased in number. Now we have superbug in Dharavi. Superbug in the sense that it can't be controlled by those antibiotics which were early used to control tuberculosis. So that means now we are thinking in terms of cocktails, that instead of going for one antibiotic, now we need 3-4 antibiotics in terms of panacea, that what would act fast and how it will act. So we do not know anything in that sense. So that is the foolishness of human species, and that is foolish thought of human species when we think in terms of this type without realizing that, without having a foresight that how it is going to impact the future and how it will change with passage of time, what we call as adaptive management. So that adaptive management is entirely lacking. Now, nutrients. So as high nutrient concentrations, they are disposed into water ecosystems, that leads to eutrophication. So eutrophication is basically increase in mineral content of water body. So when it happens, what happens over the time, that as I said earlier also, nitrate is the most common chemical contaminant, and it is responsible for blue baby syndrome. So what happens with time, when these chemicals they increase the nutrients in that water body, the algae they start growing, and the animals which are in the lower layers of that water body, they do not receive sufficient oxygen to breathe. So although it is called eutrophication, but it leads to hypoxic conditions for other organisms. Then we have extensive amount of pesticides which are used in agriculture feeds. And now we know they are carcinogenic, they are poisonous for aquatic life. That is what we know. But whenever these pesticides, they enter the food chain, that also causes human morbidity. We have a data from around 10 districts from Punjab last year regarding the usage of pesticides, that how farmers they use the pesticides, what kind of pesticides they are using, are they aware of the type of pesticides which should be used, then it was an alarming revelation that they do not know which pesticides should be used, what quantity should be used, what protective gear should be used for spraying that particular pesticide, and they were just throwing it like anything, that okay, this dose is not sufficient to control the pests, then increase the dose without knowing whether this pesticide would work on that particular insect. No, because like for example, we use HIT in our kitchen, we do not know what is the composition of it. We are using for lizards, we are using for cockroaches, we are using for mosquitoes. So whatever insect or whatever we think that it is going to harm us, we use that kind of chemical composition. But during the course of times, in terms of evolution, we never have a foresight or forethought that how it is going to impact that organism, the genome of that organism, and how that organism is going to respond. So this is a kind of never ending story. We keep on working on one side and we are destroying the other side, and nature comes up with another type of adaptations. So this is a chart that is, you can see over here, global progress in food consumption and how it has gone up. And as far as the extrapolation for 2030 is concerned, you can see well that how it has increased. And as far as the developed nations, now there's no term like developing nations or developed nations, and how this world population growth is increasing and what is going on with passage of time. With increase in population growth, there is increase in food production as well as food consumption. So given those pressures to increase more food supply, to have more food production, what is happening over the time, we are looking for pesticides, we are looking for other things, we are looking for other measures just to increase the food supply, and in order destroying the whole scenario. So this is the fertilizer consumption. You can see over here, although the data is up to 2009, that was from a paper which I took 2013. And over here at the top of slide, you can see Asia is leading. So it means this is the fertilizer consumption with Asia is showing over this graph as compared to look at Europe, East Europe, Africa, Oceania, North America, and South America. So as far as fertilizer consumption is concerned, we are at the top. And on other hand, we say that Asia is giving food to rest of the world. So at what cost? So this is the cost which we are paying, that we are using maximum fertilizers, and we are using when we are consuming or using maximum amount of fertilizers, obviously we are going to pay the price for this also. So over here also, you can say pesticides in aquatic ecosystems. As I said earlier, they are the top priority contaminants in aquatic ecosystems, and they kill almost 350,000 people each year. And even at low concentrations, they are persistent and bioaccumulate in data. So these are few examples of organisms which are in aquatic ecosystems, which are impacted by different kind of pesticides, given their mechanisms like algae, Daphnia fishes, organophosphorus pesticides. Then we have organophosphorus pesticides, other aquatic organisms, and some of these species like Daphnia magna and other aquatic organisms. Now, when they increase or they develop resistance also, on one hand, most of the population is destroyed. On the other hand, some of the surviving organisms, they develop resistance. And when they develop resistance, we call it an evolution as evolutionary rescue, because bacteria or other organisms have a tendency of horizontal gene transfer. So when we think in terms of horizontal gene transfer, these genes for resistance are easily transferred from one organism to another. So when they are transferred, on one hand, they are being killed, the ecosystem is destroyed because the chain is destroyed with the use of these pesticides. On other hand, what we are doing is we are creating those populations which are being resistant to these pesticides, and with passage of time, those populations will create problems for us also. Then salts, extreme groundwater extractions for agriculture purposes. So when we do this, that is, we are removing lot of groundwater extractions. Like for example, earlier we used to say the water is available at 100 feet or 100 meters. Now we are going to 300 meters, 400 meters. So that is, we are looking for a lot of extractions for agriculture purposes, and reason is now there is an interesting story that this leads to intrusion of saline seawater into groundwater aquifers, causes salinization in coastal areas. So whenever salinity increases, the biodiversity of microorganisms, algae, plants, and animals, obviously that declines. The reason being because due to salinity, the pH of water changes, and some of the organisms like fishes, they may not be able to adapt because there are two categories of organisms. Some we call as euryhaline, other we call as stenohaline. So some can adapt wider range of this salinization or salt tolerance, and others have strict range of tolerance for these salinization conditions. So in that sense, the organisms are impacted in different ways, and when they are impacted in different ways, the result is that whole of the aquatic ecosystems and is entirely imbalanced, and it also alters the geochemical cycles as well as decline in aquatic biodiversity. Then we have emerging pollutants, that is like antibiotics. So in this course, you might have discussed those emerging pollutants also, like antibiotics. Then we have vaccines, then growth promoters and hormones. So as far as European aquatic environment, we find that more than 970, about 1,000 emerging pollutants and their metabolites are recorded. So whenever we use these kind of antibiotics or vaccines or hormones or growth promoters, obviously they are to be metabolized inside the body. So when they are to be metabolized inside the body, having their effect on that particular organism, those metabolites also pass into those ecosystems or outside environment. So when they pass outside, the result is that they are going to enter into our food chain, into our system, into aquatic ecosystems, and that is going to create a problem for our survival also. Then emerging pesticides, they are hospitals. Emerging pollutants, basically they are from hospitals also. Then we have animal husbandries also. Then we have household discharges also. Then we have companies which are manufacturing drugs also. Then we have wastewater and sewage water treatment plants also. Then we have inputs of disposal of municipal wastes, then industrial wastes, then agricultural wastes, then excretion of pharmaceuticals, then accidental spills, which we generally do. So sewage, basically, it contributes highest amount of waste released in aquatic ecosystem, like whether it is Delhi or it is Yamuna or another rivers. This is a common scenario, especially in India. That is, whatever the sewage we produce, what we see is that if there is a river or canal nearby, we generally think let's dump into that. So when we dump that kind of sewage, it contributes highest amount of waste, and this causes extensive ecological degradation. Extensive ecological degradation, maybe in terms of pesticides, maybe in terms of heavy metals, maybe in terms of eutrophication. And this kind of ecological degradation, ultimately this leads to loss of biodiversity and severe flooding. And then we have decline in water quality and availability, because most of the diseases do occur in human beings when they have water, clean water, or water which we feel fit for human consumption, that have some sort of mixing of sewage waste. So that is the problem over there, and it has high BOD. So when it has lot of organic load, obviously to destroy that organic load, lot of oxygen is required. So this is also life-threatening for fish population. So it is life-threatening for biodiversity, and it also contains, as I said, that pathogenic organisms that result into waterborne diseases. Because I think last year also in Sangrur district of Punjab, then in other areas also, in Patiala also, there was an upsurge of waterborne diseases during rainy season. So the problem was there was a leakage of sewage with pathogenic bacteria that then they enter into our system, and then that water is being consumed by human beings. Then these are the common pathogenic bacteria which are there. All of us know these, and they generally impact our gastrointestinal tract in different ways, and some of them have become so resistant like this one, Helicobacter pylori. So this is a sort of, now I do not call it as a pathogenic bacteria. I generally call it a commensal because it has learned to live with human species because it causes lot of problems. But still, you find at least 1 out of 10 has Helicobacter pylori, and we carry that with ourselves. Then we have helminthes, different type of like Ascaris and liver fluke and different kind of other organisms, which Taenia solium tapeworm. So they cause different kind of diseases, and they are being carried by the pollution or the contamination which we do to the water bodies. Then in protozoans, we have Blantidium coli, generally it causes, then rotaviruses among viruses, then Entamoeba histolytica, then Giardia. So whenever you look at antibiotics, whatever antibiotics we prescribe for this kind of diseases which are caused by these organisms, you get a combination of 2-3 drugs. So why do you get a combination of 2-3 drugs? One is for giardiasis, other is for other kind of bacteria or other kind of pathogens, because we are not sure what kind of infection we are carrying. Because whenever we carry different kind of sets of bacteria or organisms which are pathogenic, which create problem, we are not sure that which kind of infection is there. We go for hit and trial method. Either they will prescribe something like ciprofloxacin, and then you will find that is for giardiasis, it is for something else also. So that hit and trial method, it is fine that we recover with passage of time, but obviously that hit and trial method also leads to kind of resistance which is being created by our indiscriminate use. Then we have heavy metals. So this is also a major source everywhere nowadays because they are the most toxic contaminants in aquatic ecosystems, either through natural or anthropogenic activities. Like, for example, iron. So this comes through pigments, paints, then fuels, refineries, then textile, then weathering of rocks. That is the natural activity of these heavy metals. Then we have volcanic activities, which are natural sources of these heavy metals. Then industries, agricultures, because they are rapidly rising. That is our development. In terms of development, we think that we have to promote more kind of industrialization, more kind of job-oriented approach. So with passage of time, what happens that whenever these kind of steps are taken, we do not think in terms of sustainable development. We think in terms of that we have something which is coming up, the jobs are being produced, but the result is all those anthropogenic sources, they ultimately lead to the production of these metals, heavy metals, and eventually they lead into aquatic ecosystems. So they are not removed from water bodies. So when they are not removed from water bodies, then there is accumulation in food chain, what we call as biomagnification or bioaccumulation in food chain. And then that causes considerable ecological damage because man is at the top of food chain. So the result is whatever is accumulated in a food chain, whether it is through aquatic ecosystems or it is through terrestrial ecosystems, that is to be end up inside human species. So these are highly toxic because they may lead to abnormal development of embryo or fetus. Then immune deficiency, like there are few discharges which are shown over here, like manganese and zinc, batteries, electrical pigments, paints, fertilizers. Then lead. Lead is a major heavy metal contaminant which is there in aquatic ecosystems, that is from batteries. Then we have cadmium, nickel, copper. So all those kind of metals, they are being released by human species into different. So this I discussed earlier also, that eutrophication. So eutrophication is that is human activity because this is from diffused source or non-diffuse source. So it causes algal blooms. So when algal blooms are there, which lead to high rates of photosynthesis. So when there is high rate of photosynthesis, that leads to depletion of dissolved inorganic carbon and increase in pH levels in water bodies and also hypoxic conditions. So there is a report, although this is an old paper, but recently there was another paper which has clearly sort of empirical data in that paper that it impairs the chemosensory activities or chemosensory abilities of organisms like fishes, because elevated pH can disturb whole of the system, the neurological system, as well as the chemosensory abilities of those organisms. So this is data you can see up to 2100 from 1870. That is how this blue line indicates increase in pH. That is when in case of very low anthropogenic carbon dioxide emissions to the atmosphere, and this is red is basically from high carbon dioxide. So historical change, how it has gone up to that we have, this is the pH and how it has, the pH is decreasing with passage of time, because when pH is decreasing, we have high carbon dioxide emissions from different sources, and those high carbon dioxide emissions, whether it is going, if it is going to change the pH, obviously the whole aquatic ecosystem is to be impacted. Now this is, as I said in the beginning also, plastic and microplastics. So 70 to 80% of marine litter, which is there, that consists of plastics originating from inland sources, such as plastic bags we have, then we have micro beads which are there. So they define microplastics, they are basically smaller than 5 mm in diameter and more threatening and emerging global issue. The economic impact of plastic pollution on marine natural capital has been estimated, you can see over here, 330 billion US dollars lost to the global economy. And plastics can make their way into food chain quite easily. So I read a paper day before yesterday that is published in 2021 paper. It says that first time they have reported microplastics in human placenta. So it means when it is inside placenta, those small particles of microplastics, it means it is already there inside human systems. And when it is inside placenta, it means it can easily enter the humans at the time of birth, also at the time in human species also. So these are the potential sources of microplastics. That is wastewater treatment plants. Then we have runoff from urban areas. Then we have agricultural, touristics, like we go to tourist stations and then we dispose of plastics like anything that we are moving, and we don't bother where we are dumping those plastic bags or plastic bottles. Then we have this kind of sources from industrial areas also. Then we have from shipping activities also. Then beach litter, that is a common phenomena. If you happen to visit any seashore or any beach, you will find lot of plastic being dumped over there. And whenever those high tides they come to the shore, you find that lot of plastic bottles they also come along. So this kind of beach litter is also causing a lot of problem. Then this is released last week by United Nations. This is a funding which is being given by United Nations, that is diving deep, new guidance on finance, ocean pollution, plastics, and coastal resilience. So these are the guidelines, because we do not have any option. We can't stop this kind of discharge into aquatic ecosystems. But what we can do is just a thought of preventive measures that which we can enforce, or which we can empower the agencies or the stakeholders, that this kind of problem or this kind of chaos can be controlled. So they have launched recently, this is available on their website, that is diving deep, finance, ocean pollution, and coastal resilience. In this booklet or this information, they have provided different guidelines that how can you control pollution in different areas, what can you do to control pollution, and how that finance can be generated for this purpose. Because in other countries like Malaysia, Thailand, or most of the Southeast Asia or Australia, basically they are surrounded, even South America, they are surrounded by oceans. So when they are surrounded by oceans, this kind of pollution is causing a lot of problem. And for that control, lot of money is being wasted, and it's paying heavily on the economy of those nations. So they are looking for different solutions, that how this could be controlled, or how this could be modeled, or how this could be reversed, so that it's not going to impact the organisms or the human species. Then these oil spills, this is again a major problem. Because when we design, when we think that we are running through industrial revolution or we are thinking in terms of transportation, there are multiple problems associated with transportation. And one of this is detrimental impacts on aquatic as well as terrestrial life, that is oil spills. And on other hand, one of the unaddressed concern or issue which I have seen over the years, whenever you go to some of the water body or somewhere to the canal or to the lake, what you do is there might be some motorboat and we generally say, okay, let's have a ride on that and we enjoy that. But we do not know that whenever that motorboat runs through the water body, it creates a lot of problems for the organisms over there. It creates ripples, it creates noise, it spills a bit of oil into the water body also. And what is going to happen in those organisms which are present in that water body, we never bother for that. And finally, what happens over time, these kind of situations or these kind of problems, they get accumulated. And if we are passing through that, suddenly we will throw a plastic bottle into that also. So these oil spills, although this is a different story over here, that is, they have mentioned, the data is there that 1% increase in spill size can cause damage of 1.178 million US dollars. So there are multiple sources for that, because the IPCC which I discussed yesterday, which I read yesterday and discussed during this lecture also, they have clearly indicated that United States is the major culprit. Developing nations are not the major culprit like India, Bangladesh, Pakistan, and other Southeast Asian countries. United Nations is the major culprit in creating these problems, whether that is pollution, contamination, or climate change. But still, we are the ones who are suffering at the most because we are at the receiving end. So there are multiple sources like oil tankers, then we have non-tank vessels, then we have pipelines, and by accident, some of the time, these spills or this oil is released into the oceans. And during Iran-Iraq war that went for good 80 years, 2 million barrels of oil was discharged into Arabian Gulf seawater. So this is the largest oil spill in the history, was recorded in Gulf War 1991 with the release of 4.8 million barrels of oil. And then we have some kind of bacteria also, which one of the Indian scientists, Anand Mohan Chakravarti, that which can digest crude oil. Fine, we have the remedification or we have the mitigation, but the problem is that we can get rid of that crude oil or oil spill, but the problem which it has created to the aquatic organisms or to the aquatic ecosystem, once it is shaped or once it becomes chaotic, it becomes difficult to bring it into normalcy. So these are the impacts, for example, on human health also. Now on coral reef also, there is a report coming in IUCN or United Nations that they are being bleached. Then on marine mammals also, then on seabirds also. So these are just few impacts, which then in terms of human health, you can see over here respiratory diseases, then we have dizziness, nausea, then cancers, then central nervous system. That is Malaysia, 3 lakh residents were affected due to this kind of problem, because in Malaysia or Indonesia, this kind of tourism is at the full swing. Then impact of marine mammals, that is those furred mammals, they rely on outer coats for buoyancy and warmth. And when they come in contact with oil, it flattens and adheres to their outer layer because it is a fatty layer. It gets easily adhered to that. Then hypothermia and whole of their buoyancy is impacted, and that leads to drowning of these organisms. And then we have impact on coral reefs, because coral are basically organisms which produce island-like structures, which we call reefs. So as oil is dispersed over them, it basically, those soft corals or ultimately on those coral reefs, it creates a lot of problem on them. Then what happens over there, as we feel that coral reefs are sustainable ecosystems, there's a lot of diversity. So this impacts their species diversity, abundance, evenness, then habitat structure also. Then This is an example from seabirds. Then aquaculture. So this is something we always think in terms of food production. Like, as I said, green revolution, aquaculture also, we think that aquaculture is something which is going to increase the food production, because there are plenty of reasons for promoting aquaculture. So one of the reasons is food conversion ratio. That is, you are giving less, you're getting more. Then rich in proteins. So when we say fishes are rich in proteins, there is a lot of food conversion ratio, and other organisms like prawn, because aquaculture is a broad term, it is not restricted to fishes now, it is beyond that pisciculture or rearing of fishes. So whenever we do this kind of practice in terms of rearing these organisms or domesticating these organisms in terms of aquaculture, there are multiple problems which are there, but we never think of those problems, that what kind of pollution, what kind of contamination, what kind of issues will be related to this kind of development. But unfortunately, this has practiced and it has glorified, glamorized a lot in Southeast Asia. For example, there is eutrophication of receiving waters. That is, untreated organic waste from aquaculture is almost equal to 75% of the pollution caused by other anthropogenic activities. So when we release untreated organic waste from aquaculture into water bodies, like in Bangladesh, the result is the mineral content of that water body increases. And when that mineral content of water body increases, high nutrification is there, which we call eutrophication in ecology, and that leads to increase in alteration in pH, then hypoxic conditions, algal blooms. So that is another aspect of aquaculture. Then destruction of mangrove forests. Because aquaculture, reason is that activities in deforestation activities in mangrove forest has been increased due to aquaculture, due to which niche of many birds, reptiles, and aquatic species is destroyed. This is that whenever those mangrove forests are destroyed for expanding aquaculture in regions close to oceans or other water bodies, this leads that we are expanding aquaculture at the cost of other activity or the ecological niches of many birds or reptiles. Then introduction of exotic species. Because in aquaculture, generally what they do is that if we are rearing or domesticating exotic fishes in, it has a tendency, like invasive alien species we call in ecology, invasive alien species have a tendency to outcompete those species which are present inside water bodies, so inside ecosystems. So whenever those invasive alien species invade an ecosystem, slowly they displace the native species, because they outcompete those species in terms of food, in terms of niches, in terms of ecological conditions. They are well adapted to those conditions, and same happens with species which are present in water bodies. So whenever we introduce exotic species in aquaculture, in natural ecosystems, they displace native species. So when this displays native species, eventually whole of the ecosystem or whole of the food chain is entirely damaged or it is destroyed, and this is one aspect. Generally, we say that that ecosystem or particular food chain is being destroyed, fine. But on other side, there are multiple effects which are the indirect effects of this kind of change. Those indirect effects, that there could be diseases also, there could be parasites also, there could be problems with the fecal matter also, there could be problems with eutrophication also, because whenever you introduce some sort of predator into some ecosystem from some other area, the kind of parasites or the kind of diseases it carries along, they are also introduced into our ecosystem. So this is the problem over there. Then contamination of water for consumption, because it degrades the water quality, detrimental for benthic organisms, undesirable odors. And finally, there is an outbreak of several diseases which is being caused by aquaculture. Now, this is an example. I got a picture there. You can see fishing net. That is, this is a plastic net which they have put in enveloping a cold water reef off the coast of Ireland. And this net is there, although this is meant for fish, but the problem is that it has engulfed a coral reef, water reef, and it will create a lot of problem for other organisms. Then we have radioactive substances like uranium mining. They also cause problems for aquatic ecosystems, then nuclear power plants, then production and testing of military weapons, then institutions which use radioactive materials for research and medicine, they also release this kind of waste or garbage into aquatic ecosystems. Then sometimes these contaminants are accidentally released or improperly disposed and threaten groundwater, surface water, as well as marine resources. Then nanomaterials. So when we think in terms of development, like the previous government, everywhere we were thinking of nanotechnology, nanomaterials, but now those nanomaterials, although they are meant for development, but they have also landed in aquatic ecosystems, because nanotechnology is basically a fastly growing field, and with various applications, and unfortunately aquatic ecosystems are major sinks of nanomaterials. So whatever we throw on land, it is not going to be absorbed as fast as we think. Ultimately, it has to be run off with water and it will end up in oceans. So this is the outcome of that advancement in physics or in chemistry, that is, they are nanomaterials, and we do not know how to dispose those nanomaterials, and they are leading or they are ending up in water bodies. So either they are in particulate or ionic form, basically they are toxic to the environment, and as we know, they remain in air and water for long period of time, and they settle very slowly. So there is a concern about their toxic effects on biological systems, such as structural rearrangement of biological molecules. So this is a nice depiction that how the things change, that is development in society, and what is the impact of development, that is climate change. Then we have demographic change, legislation, what we call innovations, then population growth, urbanization, and then developments in sectors, public health, food, cities, technologies. For example, generally when we design cities, like the industrialist, when they get those permits for designing a particular sort of hub or designing a particular building for that particular industry, or for some sort of computational facilities, or developing some computer spare parts or anything, they generally say that they have got the impact assessment and they are getting the permits, they can design their building over here. But the unfortunate situation is that whatever we have given the permission, we generally say that you can design a park along, you can, but we do not know what should be done with that park, what kind of trees, what kind of care should be taken for designing that park. We think that some green belt has been designed, it is fine, but ultimately what will happen, that is a kind of development without vision. So same is the situation over here, pharmaceuticals, pesticides, biopesticides, then we have emissions, and ultimately they are ending up in oceans. So over here you can see, although I'm not going to read this, that open ocean contaminants, their synthesis and assessment, like oil, human input, then hazardous, and most of them are there. So most of them, they fall in confidence level that is high, and very few are in medium as well as low. And these are the taxonomic groups mostly impacted by ocean contaminants, like human beings, marine mammals, reptiles, seabirds, fishes, invertebrates, corals, and phytoplanktons, as well as euplanktons. So they have a sort of data that what kind of impact is there. Although this empirical data, we are so poor in collecting this data, this is a bit of very, we can say, less volume of data which is there. But even this less volume of data which is being shown over this slide, it clearly indicates what kind of impact we are creating on organisms. And over here in the first slide, although this is from ocean contaminants and it is impacting humans also, radioactivity, carbon dioxide emissions, and mercury, nutrients, and I thought it would be better to think in terms of India also, because contamination and pollution of water, as far as India is concerned, how we stand now. This is a picture from Yamuna, that is poisoned waters of Yamuna, and the data is already there. Almost 38 million Indians are affected by waterborne diseases, and 1.5 million children die of diarrhea. And among children which are, or infants which are less than 5 years of age, rotavirus is one of the major factors for this kind of diarrhea. Then other bacteria are also there, but it also affects the biodiversity of freshwater fishes also. And as far as Zoological Survey of India is concerned, or World Wildlife Fund is concerned, they clearly indicate that freshwater biodiversity due to this contamination that has decreased by 84%. So what is the reason? What are the reasons? There is overexploitation, there is invasive species, then we have sand mining of rivers. Then the first and foremost leading causes habitat modification due to pollution or flow modifications. So this is already I have covered in the. Now India's dying rivers. Number of river stretches, this is what we have, 445, and number of polluted rivers, that is 275. So it means more than 50%, almost not more than 50, almost 70 to 80% are polluted. And 5% of global water carried by Indian rivers, and this is a sediment, 35% sediments we do carry. So increasing now with this, there is a concern about economy as well as the cost also. As India grows and urbanizes, our water bodies are getting toxic. 75% of surface water in India is unfit for consumption. So this is a recent report, you can read it out over here. Then that there is a release of pollution upstream, lowers economic growth in downstream areas, reducing GDP. So everything is interconnected. One small step or small casual step would lead to major problems, because ultimately it is going to affect the GDP also. Now, toxic water, toxic crops. So when we do, this is typical in Uttar Pradesh, in other regions, even in Punjab also, you can visit Ludhiana. So what farmers do, they release putrid water from a nearby canal, which is contaminated with raw sewage, industrial chemicals, then contaminants are finding their ways into India's food chain. Because when they are there into India's food supply, plants have a tendency to store these kind of chemicals. The vacuoles, they have a tendency to store these kind of chemicals, because plants, instead of excreting, generally they store these kind of chemicals. And when they store these kind of chemicals, and when we are using those vegetables and those kind of plants, obviously they are going to enter in our system, and they are going to affect our metabolism as well as our different systems. So here, this is the number of districts contaminated with contaminated water in India in 2019. Nitrate, you can see about 400 districts they are contaminated, they have contaminated water with nitrate, about 300. Then alarmingly, fluoride is also there, but you can see arsenic. So 70 districts are also carrying arsenic in their water, which is clearly unfit for human consumption. Then what kind of effect water pollution has on human health? That we know it kills. There is a study which was published in Lancet in 2015 that contaminated water can make you ill. It sickens 1 billion people. Then diseases, as I said earlier, they are different kind of diseases. They are carried by using water which is unfit for human consumption. Then in United States, even swimming can pose a risk, because they contract health issues like skin rashes, pinky eye, then respiratory infections. Then on our environment, I discussed already that eutrophication, we have dead zones, because dead zones are those zones where water is essentially devoid of life. There's no biodiversity, there are no plants or animals, so they are basically dead zones. And then we have harmful algal blooms, the product of dead zones as well as eutrophication, which can also produce neurotoxins. So this is the strategy in terms of mitigation, which they have released, although that is being revised now for 5 years. It was there by United Nations, their environment core areas where we need to work. That is, they have developmental, sustainable developmental goals. As I said, for water, they are there, but all are intermingled. All somewhere down the line, they merge with one another. So these are different goals, that how we should work together, how we should think in terms of this situation, that we can, although it is beyond control now for human species, but we can stop somewhere and we can think that how we can control it. So these are core priorities of United Nations Environment Freshwater Strategies. They are short-term goals, as I said in the beginning, that they have different sustainable development goals. They have set some Millennium Development Goals also, like 6.3, 6.6. So there are different goals which they are setting up, like meeting the global water quality change. That is how ambient water quality should be managed. Then stop countries to develop ambient water quality standards and guidelines. There should be some guidelines, we should support them. Then addressing current and emerging freshwater pollutants, global waste initiatives, GPA. Then assessment by United Nations. Then this is their 6.3. If you go to the website of United Nations, they are predominantly available on the website. These sustainable development goals, as I mentioned, there are 17 goals they have mentioned over there. And there are different other bodies which are regulating the status of freshwater ecosystems worldwide. IUCN is also there. Then we have Ramsar Convention. Then what are the values of aquatic ecosystems? What we should do. This is another sustainable development goal, 6.5. So in that, they have mentioned what we can do is like trans-boundary water management and cooperation, where two nations share the same water body, like oceans, and there they can control that kind of environment, or that kind of pollution, or that kind of contamination. Then capacity development. Then we have this another one, which is there, 11.6 and 11.5, that is promoting solutions to mitigate impacts of flood, erosion, and runoff. Then understanding the impact of polluted water ecosystems. So it is not is a sort of situation that we can focus on one thing. That is, we focus on water pollution, then we keep on thinking in terms of water pollution. In terms of mitigation, in terms of management, we should have an integrative and holistic viewpoint, that we think in terms of science as a science, not in terms of split scheme, that this is to be done by chemists, this is to be done by biologists, or this is to be done by somebody else. That way we cannot control this kind of chaos. Then humanistic solutions, like reduce your plastic consumption, or reuse or recycle whenever it is required, then properly dispose of. Then with your voice, what we can do is that there are different impacts which are there. So these are, you can see here, new generation of membranes, then Lawrence solar water pumps, maritime area integral supervision, active nutrition. Then again, this we can use. Then we can use technology for high resolution monitoring. Again, technology will be into play when we think in terms of that. That is automated geotag, time stamping, real-time sensors to gather data, that what is happening over there, what kind of pollution is there. Then we can think in terms of future, how to manage future emerging pollutants, EPA in river basins. So this is they shown over this slide. Then recommendations. So this is one of the goals of United Nations as well as IUCN for ecosystem management, that we should identify drivers of chemical pressure and predict their future development. That is adaptive management. And then we should risk reduction measures. That is, they are required to address multiple exposures to legacy, present, and emerging pollutants in a solution focused approach, horizon scanning, sector-specific analysis, substitution checks, then enlarge the capacity of integrated water management, then changes in agriculture farming. Although this is not possible, then we'll lead to change in emission of pesticides, insecticides, whatever we are using, then untreated flow, then risk and benefit assessment, then trade-offs we can think of, then production and use of sustainable chemicals should be promoted. That is the use of better degradable and less hazardous chemicals or substances. So structurally related substances should be placed together in a group. So these are the challenges. In the beginning, I said that IPCC 2022 report, which was released on 28th of February this year. So major issue is now climate change. So climate change, increasing water scarcity, population growth, these are the challenging which current human species is facing, and urbanization. And by 2025, half of the world's population will be living in water-stressed areas. So reuse of wastewater to recover water or nutrients or energy is becoming an important strategy. So increasingly, countries are using wastewater for irrigation options for, then we have to evolve those methods. We have to think, and these are a few of the goals, because this is a voice which should go ahead. Ecosystem restoration, these are the 9 goals which have been promoted by United Nations as well as by IUCN, that what we should do in terms of restoration, whether it is aquatic ecosystem or it is terrestrial ecosystem or it is a different grassland, mountain ecosystem. That is, our approach should be inclusive, it should be participatory, it should be equity in terms of gender equity also, and then the processes or whatever we are doing, it should not be like this, that today we are thinking of reducing plastic pollution from water bodies, then it's not going to help us anyway. It should be continuum of restorative activities. They should continue for a longer period, and we should aim or think in terms of highest level of recovery possible for ecosystem health and human well-being. It should not be that we should think, okay, average recovery or moderate recovery is there. The aim should be the highest recovery, then drivers of ecosystem degradation. This is the major concern. So whenever we think in terms of reversal or restoration, first we should know what are the issues or what are the problems with this particular ecosystem, whether aquatic ecosystem, whether it is pollution, or it is microplastic, or they are radioactive substances, or they are nanomaterials. So they are creating the problem. We should think in terms of that. Then all type of knowledge and promotes their exchange throughout the process. Because number of times what happens, the indigenous knowledge which is there with the local people, we generally ignore that. Whenever we visit some area, maybe wetland or aquatic water body, we think we are the scientists, we need not to talk to them because they are illiterate, they do not know anything. So that is a major problem, because most of the time we know that the indigenous knowledge which those people which live with that particular type of ecosystem, they have gained through their generations, that have passed from generation to generation, and that is sometimes much better than what we get when as scientists over there. So we need to incorporate all type of knowledge, and that should be exchanged. And this point is quite important, the 6th one, that it should be tailored to the local context. That is, the focus should be on local context, on a particular aquatic ecosystem, but you should have a larger picture or larger framework in mind, that whatever steps you are taking over there, those steps should be applicable to a larger scale or at a larger perspective or at a global scale. Then socio-ecological or cultural settings, they should be there. Then we have well-defined short and long-term goals that we are going to achieve within this period of time, this, and after long period of time, this is going to happen. Then 8th point, that is plans and undertakes monitoring, evaluation, and adaptive management. So basically, adaptive management means that whenever you're going for a long-term restoration, whatever problems or whatever you see in that restoration activity, you need to plan accordingly. It's not like that you have planned for 5 years, and if something in between happens, you're not going to change yourself. So that should be flexibility should be there, so that you can deviate or you can modify accordingly. That is adaptive management. And then finally, we should integrate policies. We should have some measures to have ensure longevity, then maintain funding and appropriable. So these are those 17 goals which I was talking about. These are 17 sustainable development goals which have been mentioned by United Nations: no poverty, zero hunger, good health and well-being. So they are trying to address this, and all those goals, although here on this slide, they have been spanned in a sense in a horizontal way. But if we think, we know that they are connected with each other in a circular fashion. So everything goes into or merges into another. That is, if there's no poverty, zero hunger, then we should have affordable, clean energy, then decent work, then inequalities should be addressed, then life on land, life below water. So if you go to the website and if the goals are there, and if you click on any of these goals, you will find a plethora of information which will provide you a lot of knowledge that what is happening and what is going to happen and how we should mitigate, how we should think of these challenges and how we should work for these challenges. For example, if you go to life below water, if you click on that, you will find that what is below water, what kind of threats are there, what kind of challenges are there, and how we should work on that. So thank you very much. And if you have any questions, please let me know.
So participants, if you have any query about this talk. Dr. Bharti has very meticulously explained the various aspects of this reason, the aspects of the environment contaminants which are polluting the aquatic life and how they are affecting it and how the aquatic life is being affected and also how it can be prevented. So we'd love to have a few queries if any participant is willing to ask. Dr. Malik?
Hello.
Yes, Malik sir.
Doctor साहब बहुत very good lecture, and just I would like to seek your comments that how this COVID virus will be varying its future in ecosystem.
Sir, actually, this is a very good question, and thank you very much. As you know, that as far as viruses are concerned, they require some medium for proliferation. So when they require some medium for proliferation, because at present, as we think in terms of Omicron, there are a lot of, the problem is again with the evolutionary biology, because now we are thinking, now we are saying that this is going to evolve, this virus is going to evolve, and then how we should control that virus. But at no point of time we are giving or paying attention that what kind of selection pressures are there, which are being imposed on this virus, and in response to those selection pressures, this virus is going to evolve. So maybe with passage of time, because most of the aquatic organisms, they are not impacted by these kind of viral diseases. Reason being, because these kind of viruses, they require lot of reservoir for proliferation. That's why when we thought of social distances or physical distancing, the purpose was not in terms of that we are thinking in terms of spreading the infection. In fact, we are reducing the reservoirs. So when we are reducing the reservoirs, obviously the virus load that is there within a person, that will automatically decrease with passage of time. So maybe in near future, some of the aquatic organisms, if they are impacted with this kind of situation or this kind of virus, then it is going to impact the human species in a very bad way.
Oh, thank you, Doctor. Thank you. Because I was reading some article that this COVID-19 virus is entering in the life cycle that in the aquatic or in the we are excreting urine and other things, that it is coming into the cycle.
No,
So
sir, yes, sir. Because earlier it also, when we thought that it has come from Wuhan, China, it eventually came, this virus found its way from the animals, and those animals are basically that have a mixed sort of lifestyle, land as well as water.
yes, sir.
So that way
Very
it is
bad.
correct that someday it may find its way, because now since the human population is there, it has got a lot of reservoir to proliferate, to multiply. So obviously with passage of time, it may go that way also.
Yes. Thank you, sir, for a nice presentation. And if any other participant has also any
Thank you, sir.
query, we
Thank you.
welcome that.
So there seems no query from any participant, and there is no message in the chat box also regarding the query. Uh, so thank you, Dr. Haminder Bharti ji, for your wholehearted, ever-present support. And we always rely on you in even in the difficult times. Thank you so much.
No problem, sir.
And you see, your expertise in the environment science has definitely added a new dimension to this course. And thank you so much. Thank you so much.
Thank you. Thank you so much, sir. My pleasure.
Okay, sir.
Okay.
Okay, sir. Thank you.
Okay, Dr. Malik. So wind up the sessions today.
Yes, yes, yes. We finished the session today.
Okay. So today had been a quite a turbulent time with the internet. So that's why I suggested tomorrow's participant also to make sure that they take their session either from the phone or from home. So, okay, sir. Have a nice day, sir.
Thank you, sir. Thank you.